Yes. Sounds good. Thank you.
Good answer. I'd like to formally request a new system. Oh, awesome.
Bridge, Nav.
Go for bridge.
Hey, can we get a ship move 9-6 meter?
Hey, Nav, do you mind just waiting a minute just so we write?
Oh yeah, just belay that. Let's just give us a second.
All right, we will be standing by.
Thank you.
All right,
Sorry, I
next
thought
time,
you were done with your heading.
I know, we're almost good. We're getting there. I just want to get my bias looking real good before we start trailing, and then
Is it still
sure.
so your Z bias? So you're going for a delta of 10 or 15?
A little less than that. So when we get deeper, the two depth sensors kind of like get more separate, I guess you could say, because like the Sirius depth sensor and D2 aren't like exact all the time. So that's why when we go deeper, we have to have a higher delta as we go down. But at first, it should be around like 5 to 10. And then since I just changed like my thrust outwards, I'm just gonna give it some time to like regulate, so we know that like, okay, my thrust outwards good. I have enough vert to keep pushing down. And then when that's all good, then when the ship starts moving and we kind of trail behind it a little bit, everything shouldn't be too big of a change. Should just be like little adjustments here and there. So that's the plan.
Because your delta Z is your distance between the two vehicles.
Exactly. Yep. Yep.
Depth-wise.
Yep. Yep.
Your
So we
Z
always
bias though.
Yep. So Z bias is just like the number, the percent of thrust that you dial in. And then your delta Z is like the actual difference between the two. So like my Z bias right now is like 76%. So that means I'm doing 76% down. So that's why it's like negative 76.
Okay.
I mean, it's a little higher, but. And then delta Z is from here to Sirius. So you never want this to go negative in this case. So negative as you're going, that means Sirius is below you.
Because then you can wrap the tether.
Exactly. Okay. Yep. Yep.
Okay. Yeah, I just didn't totally. So you're managing your percentage of thrust.
Yep.
To match and kind of increase your Z or increase it.
To match the winch. Yep.
Yeah.
And then like as we move and everything, that kind of like changes because now like we're dragging Sirius and D2. So sometimes you need like to put more down thrust or. Sometimes you need to like push out more so that the streaming stays, the headings stay the way they should, inverted of each other. But it's looking pretty good. So let me check with Copilot, and I think we can get moving here. Copilot, how do you feel about a move?
Hey, yeah, that's good.
All right, Nav,
good.
go for it. Thank you. Yep.
Bridge, Nav.
Go ahead.
Sorry about that. We're ready for a move. Range 96 meter, bearing 137, speed 0.2 knots.
Roger. I've got 96 meters, bearing 137, speed 0.2 knots.
That's correct.
I'll let you know when we're there.
Pilot, did you log about
Hey. Log about what?
these guys here?
No, I did not log about
Oh
it.
my goodness.
I asked a question about it though. Let's
You
see.
did.
Monitor buttons, no work. How's that?
Perfect. Sounds good.
Nice. Ship's moving ahead.
How do you feel about cycling that relay with aft cam on it?
I can't. Oh, the flex link. I can't
Yeah.
cycle that. Yeah.
Or I was on
Oh, let me get
on
let me
D2.
get another photo of DC bus though.
Oh, okay. Yeah, it's 122.
Typically every 500 meters, unless I see like a big drop, and then I grab another one. Oh god. Okay. Oh, I took it with the pilot one though. Alright, Delta's looking good. Comps look like they're doing what they should. DC bus is doing its thing. Fiber values. What do we got? Those look like what we've been seeing, so it looks good. Multi-beams pointed down and ranged out. Same with Cirrus sonar. Cirrus cameras pointed all the way down. Winch. Ooh, it's a little hot on winch now. Back down a little bit. Okay, Pat, I came a little hair off winch.
Alright, hair off winch.
May or may not affect you.
Okay. Oh, max was 35. It can't be that bad. Right?
Oh, it wasn't bad, but
Seen worse.
yeah. Yep.
Nav, bridge.
Nav?
Hey, just wondering what our ETA is and our predicted depth.
So we have 2 hours until bottom. Or correction, we have 3 hours until bottom with a target depth of 5370 meters.
Bridge copy is 5370 and 3 hours. Also giving you an update that we have completed our ship move at this time. And Nav, I'd like to come over 10 degrees to the left on my heading, just to get the DP to hold a little nicer. Is that okay with you?
You guys okay with 10 degrees?
Yeah.
Yep, that's fine.
Okay, thank you.
pilot. Is that you just cycling? Copy. Understood. So Nav, let's see, landing flat bottom and then trying to find some slope. Is that the goal for today?
It is. When Chris comes up, we might push that a little bit further over more. So
Copy
we might come closer to the slope here to start, but he's going to make that determination probably around lunch.
Copy that. All right, vehicles is looking good. Streaming north. They look nice and happy. Yeah. No, that's a good perspective you got. There you go. All right, so I got the suction jar. Ran all the air out. Run jar 6, the bypass. That looks good. Do you have a light out? Oh, sorry, you're talking. I wasn't listening. Okay. I think you're rubbing off on me. I think you're... No, we can keep it on. If you want to cycle it to look for ground faults, you can cycle it. Typically, we want to keep it on, though. take one. One of the two rotos, yeah.
Sean, can you hear me?
Yeah, you're a little quiet, but I can hear you.
Okay, never mind. I just saw the two.
Oh, you're good, actually. I just turned you up a little.
I just saw the two oil comps came back. They were.
Oh, yeah. Sarah was, she's ground fault hunting.
Got it.
So she cycled those. It's very good eye, good eye. It's not it, though. Yeah. We have time if we're going to use it. That'd be a new one. I don't think that we've ever had. Those things just kind of work. I don't think they've ever been an issue. Oh, the R3 camera. I guess you could put the R3 camera. I could put suction jars. Let's see. Over here. And then we could do, is that 12? You still cam? Oh, we took it off. Ah, duh. Silly me. Okay. Oh, that's what it was. Yeah, yeah. I'll put it back to jar. I believe so, yeah. asked Michael to cycle the switch last night. So I didn't reset it from this screen, but if you cycle the main switch for the winch, it resets all the monitors and it resets that value, which makes, then you can reset all three at once. Otherwise you would have to, I tried again to do it from the screen and it still freezes it up, so yeah, it gets unhappy. So I think after each deep dive, just cycle that switch downstairs and it'll freshen us up for the next day. I did a few days ago. No smoking gun, huh? I think so, yeah. Oh, we're using one of them. Alright. Well, don't touch stuff you don't know what it is. Well, good, we can look at it then. Yeah, now that it's off. Actually, it looks like it came up. from 58 to 66. There's a little something there. 69. Can write it down. Call it a 10 KO. Nah. I'll survive. You got any pocket bars? Jolly Ranchers. Yikes. That's how they get you. Did you go for the smooth or the crunchy? Nice. That's nice. I never got into the combination of jelly and peanut butter. I like them separate. Yeah, I was a weird kid. Still am. Ah. Yeah, they sell, what was it, Yellowstone? All the national parks in that area. They sell like everything huckleberry in all the stores. They're super pumped about huckleberries. Yeah, it's not bad. From the Paro? Oh, Sirius, Sirius Power. Yeah. Which looks, it's been looking typical. I don't check it as often as D2. Just less things, so it's easier to keep track of in maintenance. Whereas D2 has a lot of things, so. Yep. think so. So we're not gonna see bottom, right? But this doesn't affect us. But if the tether was being more happy with the clockwise turn, this is where you would have to like do a 3/4 turn to maintain that, as opposed to the shorter port turn. Something to think about for setup. Yeah. Levi said it became with a port turn. Yeah, with a counter turn, counterclockwise turn, it became unhappy. So you'd have to do a larger, yeah. Sounds good. Tell me more. Oh, like the fake goop? No, I don't like that. What? No. I'd rather have like a real marshmallow. Why would I want the sludge? I guess my answer's no. Not a fan. I think you should just try a plain peanut butter sandwich. I get that. I could try some huckleberry and go from there, one step at a time. I don't want to move too quick, you know? Yeah, it was easy. That's true. I would have just eaten that, yeah. Hey, watch it.
What's going on? Yep.
Morning.
Looks like 2 hours from now. Yep, 11:30.
That's something as a kid I loved, and as an adult, now I'm like, oh, I remember as a kid loving it, but now it's like, I don't know if I could do that again. Yikes, that's a psychopath. Woof. I'm sure she's very nice. Sorry, I got a little hot there. I apologize. She listening? Oh. I'm just.
You brought 12 pounds of cheese. To where? Like just domestic? Should have wrapped them in lots of saran wrap. Oh. Was it special cheese? Damn, that sounds good.
got eyes on the bottom.
Ooh, I like the view. Looks promising. The ROVs are getting settled on the seafloor. And my lovely scientists in the back row are getting settled as well.
All right, kia ora, ladies and gentlemen. I'm Chloe Anirag from the Cook Island Seabed Minerals Authority, and we are coming to you live from the west of our Cook Islands exclusive economic zone. And we have just touched down for our 12th dive on this cruise at depths of about 5,300 meters. Now I'm going to allow my colleagues to introduce themselves, and we'll start off with Dr. Amanda.
Kia ora, everyone. Thanks, Chloe. Great to get a chance to dive with you again today. It's been a great mission so far. Appreciate it. Really have enjoyed these dives in your waters, getting to explore your backyard. My name is Amanda Demopoulos, and I work for the U.S. Geological Survey. I'm a research ecologist based in Gainesville, Florida, at the Wetland and Aquatic Research Center. And beside me, I have Rima Brown. Say hi, Rima.
Thanks, Amanda. Kia orana, everyone. My name is Rima Brown, and I also work with Chloe at the Cook Islands Seabed Minerals Authority. And it's great to be here today, and I'm looking forward to all the exciting things that we'll see, especially the sea cucumbers.
Calling all sea cucumbers. And last but not least.
Well, thanks for the not least there, Amanda. I'm Dr. John Parianos, and I've the pleasure and privilege of working with Chloe and Rima at the Cook Islands Seabed Minerals Authority. And we're also looking forward to hopefully what will be a fruitful day's traverse along the seabed here. Looking to see what, clearly we're going to see a few nodules, but clearly we're interested to see what animals we do find associated with these nodules. This part of the Cook Islands has only been dived once before, and that was yesterday. So this is really going to be a voyage of discovery for the team on the Okeanos. And for all of you on shore who have dialed in or watching or contributing on the science chat, welcome as well. And we're really looking forward to your support.
Thanks, John. I think we're just waiting for the green light from the ROV. Looks like they're getting settled. And once we start getting underway, they're going to turn the lasers on, and we'll start making way. I guess we're going up slope, John. Is that the plan for the dive?
Come again there?
What's the game plan for this dive?
Yep, we're going up slope, so we're on a small plain, volcanic plain, and it tilts very gently to the east, and on the western side is a cliff. We're not going to make it to the cliff today, and that's what we call, it's like a fault cliff there on the seabed. If you see there in front of us, you can see a shark tooth. Right there, center left. We're going to probably go straight over that. They're fairly common down here at these depths. And we'll work our way up this slope gently. And it might be nodules all the way, or it might change. Often things do change in this part of the world.
Watch lead to pilot when you're ready. Hey, can we just get the lasers on, please?
Thank you.
Awesome, thank you.
Yeah, I think it looks like a sea cucumber on the left.
Thank you.
And then there's a lot of these little tiny white dots. I'm curious what those are. It's a chilly 1.2 degrees C down here. And we're about 5,360 meters. And then looks like a larger, maybe rock on the left. It seems like a different object compared to all of the nodules that we're seeing. Yeah, maybe. What do we got here, folks? Nice.
Nice.
sea pig.
Yay!
He looks full.
It does look full. Yeah, this Elpidiid sea cucumber. Rima conjured it up for us, wishing for sea cucumbers, and we got one right at the beginning of the dive.
Good.
You can see the coiled guts are full of sediment that they eat and pick up off the seafloor. It's a great view of the nodules too.
Wow, look at that.
It's interesting, the sea cucumber kind of looks fuzzy, like it's
Yeah,
got
it does.
like fur.
Yeah.
Do you know why that is, Amanda? Is this a different
Uh,
species of?
I don't know if it's a different variety of what we've seen for holothurians, but yeah, it looks like they've got tiny spicules kind of jutting out from the body form itself, sort of spines. Really great shot of this holothurian or sea cucumber walking along the seafloor and feeding. Thank you. Thanks, pilot. And we got a good shot of the nodules as well. Chloe, it seems like they're like irregular in shape.
Yeah.
Some of them are more rounded, and
They seem generally more rounded, but definitely irregular in shape. And this is in stark contrast to the area we dived yesterday.
the chat, the shoreside scientists are remarking about the fuzzy pig. The sea cucumber that we were just imaging is very fuzzy. We haven't seen it before.
So how far is this dive site from yesterday's, Amanda?
Oh, that's a good question. Maybe we can ask John, since he's helped plan the dive today.
Let me consult my computer quickly here.
There we go.
We're not very far away. I think I've got it on this side here.
Yeah.
So we would be if you wanted it in kilometers, we would be about 17 kilometers away to the southwest,
Interesting.
which is around, for the more nautically minded people here, around 10 nautical miles or so. We wanted to dive the abyssal plains, and we were using all the data we had at our disposal at different times of the planning process. And when we got our eye in, so to speak, on the no nodules we saw yesterday versus what we call the backscatter response from our multi-beam system, we were able to pick this site as a place to go today. And what we really wanted to do was have one site with nodules and one site without, to compare and contrast the biology that we find on those two sites as best we could. So obviously the type of biology you find in a particular site is dependent in part on the substrate that's available. It's also dependent on things like food availability and water depth, etc. And these two sites are very similar depths, very close together, but of course very different substrate available for life to borrow from, to attach themselves to. And you can see there's a good example of one there right in front of us. We might go and have a look at that. And who wants to guess what that is?
Can we zoom in on
Sponge.
that pilot? Thank you.
And the sponge wins, I think.
It looks
yeah.
like it.
Yeah, glass
Good.
sponge.
Good job, Rima.
Yay, Rima.
Yeah, that looks like a glass sponge. one of our earlier dives, Amanda, we saw them. One of our dives, I think it was on a seamount, was quite a significant amount of glass sponges.
Yeah.
Yeah.
They somehow make a living in these deep sea environments where they can trap food, pump the food water through their bodies, through their pore spaces, and collect food. They also have a lot of different animals that live on their stocks, and so they're kind of like a condominium in the deep sea. They're a little apartment building for home, a home for lots of things.
I love that description.
Yeah.
I'm gonna remember that.
Oh yeah.
Yeah, we saw quite a lot of sponges on our cruise last year, and that was more southeast of the Cook Islands.
Oh yeah.
Both on the abyssal plains and the seamounts.
Oh, interesting. Yeah, and we have been able to successfully sample a glass sponge.
That's right.
And it's currently sitting in our lab. We're very excited to take it back with us.
We're not sure how the sponge feels about that, but yeah.
Oh, there's something hanging out on the edge of the sponge here.
Potentially, and
Okay, that's a good video.
Below the sponge you can see some lines on some of the nodules there, like little tunnels or little tendrils, and a lot of those foraminifera xenophyophores that are a special kind of, maybe you can describe them better than me, Amanda, a single-cellular organism that agglutinates sediment onto them.
Very good.
Yeah.
Very good. Yeah, the sediment kind of gets trapped in their tests and their forms, and then likewise, like the sponges, the xenophyophores can also serve as a home base for a lot of different animals that we can't see. There's a fair amount of different kinds of xenophyophores that we've encountered on the dive so far. Some of them are more planar, sort of flat, and they almost look like mushrooms growing on the seafloor, but they're not. And then we've seen a few different types of forams living on the seabed, which create calcareous tests, so hard parts made out of calcium carbonate. Quite a variety. Some of the nodules themselves look like they're dusted with fine coating of sediments, and others look a little cleaner. And that may be due to some of the animals that we've been seeing, that kind of clean surfaces. The darker surfaces may be due to the sea cucumbers and other animals that come and feed as deposit feeders, consuming sediment as they move along the seafloor, keeping the seabed tidy.
So for those of you who are curious about where we're located right now, we're in the northwestern part of the Cook Islands. This area is called the Samoan Basin because one of the closest land masses to us is American Samoa, and of course Samoa itself. We're located about 100 nautical miles south-southwest of the island of Puka Puka and its nearby island of Nassau, two of the islands of the Cook Islands.
Gastropod.
Yeah.
Gastropod.
Oh, so cute.
We've seen this type of sea snail or gastropod on previous dives this series, as well as when we were out here on the Nautilus last year. It's interesting, not every single white object that we zoom in on the seafloor is the same thing. So every time we zoom in, we might see something a little different. These images will be reviewed by our taxonomists back home. Hopefully we can get an idea of what we're looking at.
And it's certainly a tough life for that little gastropod, because not only are we quite deep at high pressures, it's quite cold at about 1 degree centigrade, and there's no light. At these depths, actually, the carbon dioxide in the water is quite a strong acid. So the gastropod has a shell made out of the same thing as your bones, which is calcium carbonate. And yeah, it's under acid attack the whole time. So it's got to make sure that it keeps replenishing its shell under these harsh conditions.
Is that a coral or?
I'm leaning towards sponge, but I will reserve judgment.
If you're wrong, Amanda, it's just recorded for the record. That's
Oh
it.
yeah,
Well,
I'm wrong a lot. It's okay. I'm comfortable with that. Yeah, it looks like like one of those carnivorous sponges.
Oh, that's what you were looking for, was it?
Yeah.
oh yeah.
Nice.
Well done.
Yep, you can see the axis of the sponge is bending over, and there are some animals living on that major stalk, including amphipods. And we're taking a closer look at the sponge itself. That's a type of carnivorous sponge. It has these spines that have hooks embedded in it, and they help trap animals, which they consume. But those amphipods are making a good living on the main axis, or capturing food as it's floating by too. And maybe keeping the sponge
That could
clean.
be the reason why this sponge is leaning.
Yeah, a little too much weight. Sort of like a teeter-totter. Nice. I'm going to zoom in on these amphipods that are hanging out on the sponge itself. You can see them feeding too, and see the white eye spots. Yeah, whenever we're good front row with this, whenever you guys are ready. Thanks. So we were able to collect a relative of this carnivorous sponge on one of the previous dives, and it had an anemone attached to it. There's a commensal relationship with the anemone. They live together. Anemone residing on the sponge as a home. There's a little shark tooth in the distance behind the sponge as we take off and get a move on.
As we get folks tuning in, we just want to say a big kirana to anyone tuning in from shore here in the Cook Islands. And we have a special shout out to Amanda. Someone from the Seabed Minerals Authority says, "Hi, Amanda." And she says she misses you already.
Aww.
That's from TJ.
TJ, we miss you too. We love having you out here. Love all your stories and great commentary, and wish you could be here chatting with us.
Hi TJ,
Hi,
Rima
TJ.
here. I miss you too.
Of course, we all miss you, TJ. but there's only 4 seats at this bench, so you'll have to watch from your seat in the office.
Yeah, this is quite different terrain to where we are yesterday. It's quite different terrain to where we were yesterday. Yesterday we saw the terrain was fine silt sediment, and it was quite loose, wasn't it, Amanda?
It was,
It's
yeah.
really, yeah. The slightest disturbance really kicked up the plumes, and we managed to get 2 push cores from there.
Yeah.
And you were saying the sediment was sloppy.
It was really, really soupy, kind of, like I say, it's fluidized, you know, super easy to disturb, and a really red color. When it came back on deck, it was red-brown in color, pretty indicative of this area and these depths. Nice. Have you guys noticed a current down here, front row? Any kind of appreciable current? Okay, thanks. Yep. We've been seeing a fair number of shark's teeth on the dive so far.
And another sea cucumber.
And a sea cucumber.
Yay.
Yay.
That's 2.
Another shark tooth.
Oh, yeah, those as well. Yeah, this is a very high dense nodule area we're in right now.
Yeah, what we can see in the survey data that we've been looking at is that there's likely influenced by currents on the seabed in the region. So there's places where there's fine, it's technically called clay minerals. These red, brown clay minerals are more likely to be maybe accumulating from the currents, and places where they're likely to be removed. So this is a gentle slope facing east, and I guess there's a chance that current is coming over the cliff further to the west, as currents sometimes do, and maybe it's running down this slope and helping clear some of the sediment away and allowing the nodules to grow well.
We're hovering around 5,360 meters at depth right now. And we're about 10 nautical miles southeast of where we were at yesterday's dive site.
Ia mai ke kai mai. E nā la la li ia pau I hanau ia I ka pō I ka lahiki kū. Ia mai ke kai mai. E nā vēlaua li ia pau I hanau ia I ka pō I ka lahiki kū. Ia mai ke kai mai. E nā puali ia pau I hanau ia I ka po I ka lahiki ku. E ola, a kau, a kaniko'o, a palalauhala, a haumaka I ole, a kolopupu, a hiki ka pua ane ane.
I think it's one of those gastropods.
They're quite rare, I think, aren't they, at these depths?
Gotta be another holothurian hanging out somewhere here. I can see the tracks.
That's right.
Yeah.
The evidence.
That's right. Do you want to talk about how the holothurians feed and then lift off when they're done, Rima?
I don't actually know much about that, John.
Oh.
Do you want to tell us more about it?
Well, you saw it on NA176. So what the holothurians do is they move along the seabed, filter feeding as they go, picking up with their, what are they called, modified hands, or the hands, the mouths are modified to be feet, one away the other.
Modified tube feet. They deposit feed. They feed on deposits. Yeah.
That's it.
Yeah.
And then when they're almost perfectly neutrally buoyant, I think, the holothurians, and then when their stomachs are full, they get heavier and heavier because they're full of sand.
Sort of like when we eat too much at lunchtime, we feel heavy.
A lot like that. And then when they're completely full, they empty themselves, and then they often lift off into the water. They start flapping around and swimming, and they rely on, I guess, a bit of, because it's completely dark and they can't see where they're going, I guess they're relying on luck to put them back down in a place that hasn't been fed before. And then they start and repeat that process again, which is why you see the tracks in strange directions and suddenly stopping. And often they stop right where you can see the casts of the holothurian who finished off.
Yeah, it looks like another one.
Must be a lot of full sea cucumbers around here because I haven't seen any of them floating.
It's lunchtime, man. They're all full still.
They're still eating.
Yeah.
There is something to look at just below the lasers front row. Pilot, if we can look at the sort of white object below the lasers while we're set down. Oh, this is something different.
Ooh, this is really interesting. Oh. Okay.
Oh, this might be collection worthy, pilot, if we are able.
It looks like the drawing of a star.
Yeah.
How beautiful.
What is this? What is this, Amanda?
Looks like an anemone to me. It's in the phylum Nidaria, but I can't quite make out, how it's attached. But anyway, worth a closer look, and it's on the small side, so not sure if they can slurp it or if we need to scoop it. So that's going to be for ROV to figure out, I think, if it's possible. It seems kind of small, so maybe we could slurp it. And we have a number of scientists on the shore side, taxonomists who've been tuning in throughout the mission and providing us with some guidance on what we're seeing. And we'll collect and subsample this for identification, and they'll be able to sequence the DNA as well.
And we're about to collect our first sample for this dive.
That'll be the first sample, yeah. Hi, pilot, or 100% sure. pretending I'm Chloe right now.
You're doing great, sweetie.
Yeah, I don't know if we can get another perspective or image from the mini Zeus front row, too. Yesterday we were able to collect a sea pen, as well as an urchin from our collections on the abyssal plain, our sediment-laden dive. That was really amazing. Yes, we can.
What do you think this is?
So right now our pilots are just getting their sampling tools ready. To collect what we think is an anemone.
Are you guys going to give me time to set up these recorders?
It's interesting, on earlier dives we did on the series, we saw what looked like a cup coral that was sort of similar in shape, but we didn't get a chance to take a close look at it. Anyway, we'll be able to collect this animal. To me, resembles an anemone, and hopefully get a further identification. I'm not sure if it's in the APSMA catalog or not, actually.
Let me look that up.
Might have seen it before.
Hey Amanda.
Hey Chloe. You think you found it? Oh, interesting. Yeah, there's some previous anemones captured, images captured of anemones in the Clarion-Clipperton fracture zone. So
Possibly. Did we see
other abyssal environments.
how many tentacles it had? Oh, let's see if we can count the number of. I'm bullpocking it, but it definitely looks like it has more than 8 tentacles.
I'm not sure. The nodule. Oh, I don't.
Yeah. I'm pretty sure the sucker picked it up, but where it went.
Okay.
Yeah, I'll wait for the...
And it went into went into jar 2. Hang on. Front row, do we know which suction container it went into, potentially? I think it might still be attached to the nodule. Yeah, if you zoom in on the nodule right below the scoop or suction, I can see what I think are tentacles. The little white.
Oh yeah, I'll just wait. Oh no, I mean, but if they did collect it, then it's, yeah. Okay, start again.
While we're waiting for this to proceed, we can notice that the... Yep.
I'm 100% okay.
We can see that the plume from today is different to yesterday. You see it settles a lot faster. Even the color of the sediment's different. I don't know if they've adjusted the cameras, but...
Some materials going into chamber number... And Levi, that went into 1 or 2? Number 1. Okay. Okay. And then you can put with nodules. Yeah. Then we know we've got a 2 for 1. Okay. We'll be on the lookout for it when we get it back on deck. Thanks, front row. That was great.
Looks like some water.
Video on our camera 3 view, is it possible to put the pilot GUI as one of the quad views? I can't see, sorry if it's on there. Video, this is watch lead. Hey, would it be possible to put camera, let's see, GUI for the pilot in the quad view? It doesn't matter, it's just shoreside is interested in being able to see the depth now and then. Thank you, sir. bio. Number 1B. Good copy. I like the team effort up there, making it happen. So I was starting to take note of some of our potential future collections to target. I think we've been seeing the gastropod as well as the carnivorous sponge. So I guess next time we see one, maybe we can get a collection.
Yep.
Do you all have some interest in a nodule collection or more sediments or both?
We've got a couple of nodules from that inadvertent biological sample.
Yeah.
But I guess, yeah, later on, let's towards the end of the dive, let's go for the piston cores again, and then let's see if we can get a scoop of nodules.
The push cores, yeah.
Yeah,
Okay.
and just a scoop of nodules somewhere. Yeah.
Roger that. Sounds good.
Probably the bigger the better. In terms of a bigger nodule, will tell you a little bit more about the history because it's longer lived. Yeah, but
Not yet though, Levi. We're gonna try to do the coring towards the end. And then I see something ahead of us that's sort of white and spiky.
It's got propellers. Just asking on behalf, if we see a headless chicken, can we sample?
Okay,
Yep.
we can try. Yeah.
all right.
Get a sea cucumber in our collection baskets. Sounds good.
We saw quite a few last year, but we haven't seen any
Not so far.
on
Yeah.
this trip,
Are
including leg one.
they all red? We saw that really dark red one.
Red
They're-
and yeah.
I've seen
all
them
the headless ones are
purple
red.
color. I'm not really sure if there's a huge variation in color, but I think that group is the Anipniastes holothurians, if I remember correctly. The headless sea chicken
monster.
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 I'm curious who actually named that animal with that common name.
We need a bottle brush for the next dive. So while you're waiting for this operation to take place, the clay in this part of the seabed is predominantly, oh, look at that swimming past.
Oh,
Who is that?
it looks like it might be an isopod, but it's not really active.
That might have been what I thought was a magna pinna when I was tuning in.
Oh, okay.
We could try and
Yeah,
suck
I
that up
think
with
it's
the
an
sucker that's full of
isopod.
mud.
It looks familiar.
Yeah.
That's what we thought, yeah.
Just
Moonopsid or something.
Just casually floating
Yep,
by.
hanging out, saying hello. Says, hi, Rima.
Oh, it's turning around. Is it coming back to us?
It's like it's falling in slow motion.
Don't go towards the light.
It's going to swim straight in the bio box.
Yeah, right.
Oh, That's great.
Nice.
Run, Forrest, run.
Oops, type of isopod swimming in the water column, giving us a nice little show. That's great.
Okay. I think we've got that one.
Thanks, folks. Yeah, and Dr. Jill Burke from USGS tuned in and commented that it was a monopsid isopod. We've seen them swimming around before, and also on these abyssal dives, so every now and then we get a closer look. Isopods are a type of crustaceans. We find some of their benthic dwellers, the ones that live in the sediment or on the sediment as well, some family members that are relatives. So we'll just keep taking a look, wandering along the seafloor, seeing what we find.
It's like mud you can't get over the bottom of your shoe.
It's stuck.
Yeah. Yeah, well, seabed mud is often highly cohesive, so instruments that interact with seabed often need to have water jets to clean them. Usually ROVs are fine because, of course, they're above the mud, but looks like we got a little bit of sticky mud when we picked up that anemone. Yeah, the mud's quite interesting if you're a geologist or a sedimentologist. It's composed entirely of minerals, mostly brought in by the wind or carried on the seas. So from volcanic eruptions and blowing off deserts. So when they analyze them, they often find a very wide range of minerals from all over the world. Another feature of the muds is that they're very high in water content, of course, because they have settled on the seabed in the water column. So their density is only slightly higher than that of water. Water's got a density of 1, and these muds are often got a bulk density of about 1.2, 1.25. That's why some of the particles drift about. When they're stirred up, they do settle. Some more, most of it very quickly, and some of it takes a bit longer. And part of the thing that helps in the settling is the pieces of clay are actually attracted to each other. They basically agglomerate or pull together from just from small electrical forces on the surface of the clay particles. That's why when we stir up the mud, taking a sample, often within a few seconds or a minute or so, the water's clear again.
Rima, you led a box coring cruise out in these waters. Isn't that correct?
Yeah, we had the Women in Science expedition a couple years ago, where we took about 15 trainees out to sea to conduct some environmental sampling, which included box coring.
That's fun. What's a box core do?
So it takes a sample of the seabed, and you're able to bring that up and sort of have an idea or a snapshot of what the seabed looks like. And then you can look at it in terms of its geology, if you collect anything, otherwise also the biology. So for that cruise in particular, we were looking at the macrofauna, was it, John? Yeah, it was a great cruise. maybe to the trainees. I'm not so sure about the advisors.
Oh. Yeah, we kind of turned the plot around, didn't we? We let the trainees do a lot of the detailed planning and execution of the tasks, and the advisors had to sit back and help from a distance a bit. And it was, there was, I think, 5 advisors, wasn't there? 5 advisors and 17 trainees.
Yeah, about that.
That sounds like fun. Box scoring at these depths is pretty tricky. Yeah, doesn't always come up with a sample.
No, that's true. We were fortunate that we were able to use a vessel that's based in the Cook Islands that had a very experienced crew on board. So the actual mechanics of running the equipment was done by the crew, and but the moment it came on deck and the box was separated from the frame, it was up to the students, right, Chloe? You were on board as well.
I sure was.
Did you guys get to learn how to deploy and recover the box score, or was that handled by the ship's crew?
No, the ship's crew handles all of that.
Oh, yeah.
But for science, the most you can get up to is just do the final checks. Everything is in place, yeah.
I think one of the students was turning on the cameras, making sure the equipment was working. They assisted in downloading of the video data and the CTD data at the end of it. They also did some of the final checks for the safety pool to make sure the box score was armed and ready to go.
So Rima, how did you guys select stations? Do you remember where to do your box scoring?
I think we didn't have a lot of mapping for the areas that we went to, so it was sort of just pick a spot. But we were looking for deep abyssal plains.
Okay.
Because we had intended to do ROV dives as well.
Oh, right on.
Yeah.
You want to talk about what happened there? I think you should have. Rima was chief scientist actually for the cruise.
Yeah.
And we had to pivot, didn't we, when we developed a problem with the ROV.
Yeah, unfortunately, sometimes things don't always go your way when you're out at sea. And we had a technical issue with the ROV, so we had to quickly pivot. And in the end, we did a makeshift, what would you call it? Benthic?
Yeah,
camera.
Oh, nice.
Out of the multi-core frame.
Okay, right on.
So we managed to do 2 of those. And we
there's
saw a
something
lot
in
of the fish that we're seeing
Yeah, look at
in
that.
these dives as well.
Oh,
Cusk
that's neat.
eels,
Some common ground there.
amphipods and shrimp. Yeah.
Awesome.
We're seeing something interesting in the center of our screen, just below the lasers.
What do you think it is, Chloe?
Oh, I reckon another holothurian. We're going 4 for 4.
Maybe it's one of those spiky, fuzzy ones.
Ooh, yes.
Those are cool.
Our pilots are slowly getting settled as we approach the holothurian.
Get to take a closer look.
You can see the tracks they've left behind to the left of the screen.
Tracks?
Waste.
Or something.
Or something.
If all that cast came out of that holothurian, that's
He'd be
impressive.
swimming.
Oh. Okay. It didn't come from this holothurian, folks. Ooh, is it a holothurian?
I don't know.
I don't know
take
either.
a closer look. Looks kind of like maybe an acorn worm. Yeah, Rima, you got it. And tarpnoost.
Oh, very.
Acorn worm. They also deposit feed, so
It looks so beautiful.
consuming the sediment off the seafloor.
Yeah.
And then you can also see their guts.
Oh,
full
we
of
sure
sediment.
can.
Did we see the size of lasers on that? I didn't see
I
it.
don't know.
I think
Maybe
it's
when we
super
come back
small
out.
because we zoomed out or zoomed in.
Yeah,
Yeah.
those nodules are between 1 and 3 centimeters across.
So it's pretty small.
So yeah, the bigger one on the right will be 3 centimeters, and yeah, 1 to 2 on the left.
Thanks.
So yeah, that fellow must be about 4 centimeters long. We can test me with the lasers when we zoom out.
That's a great shot of the
Nice.
acorn
Great
worm.
video.
Nice shot. Pretty small.
I'm listening. Yeah. Oh. Okay.
Oh.
We're always interested.
They're always interested. Little
Let's
dots.
go for it. Ooh, maybe. Yeah. Interesting.
Oh yeah, quite a collection of white objects.
Yeah, even in the distance.
Yeah.
Few mounds. Oh, what's that?
What are we gonna find out? Oh.
Ooh,
Oh, I think it
gastropod?
might be our target.
Oh.
Are we in a good position
Yeah.
to collect front row, potentially? Okay. This is a sea snail gastropod. We've been seeing this on this dive so far. Dr. Chris Ma from the Smithsonian identified it as a margaritid gastropod. That's the family. And we'll, yes, please, pilot. Yeah.
Love it.
And that may be of the genus Gaza, I guess. I'll have to take a closer. Yes, this would be a biological collection. I'm thinking we should slurp it if possible into a different suction chamber. And I heard the pilots, I was eavesdropping, they said that there's a little less than 3 hours left on bottom. So we've got a bit more time. No, it's two. The first one was the anemone video. So 2B. Correct video, 2B. It's neat. You can see the suction chamber rotate on the GUI.
Yeah. Do you want to explain how the suction tube works, Amanda, through to the containers?
I can, sure. The suction hose is something that the ROV manipulator or JAWS will grab. Pilot navigates the end of the hose over to the sample that we want to collect, in this case the gastropod. And it is like a vacuum cleaner. And the pilots can increase or decrease the speed of the vacuum according to how much they want to basically vacuum and suck up. And so in this case, they'll try to gently vacuum up the gastropod into a collection chamber on the vehicle, and then it'll be contained for the rest of the dive. In addition to the animal we'll get some sediment and possibly some nodules like we saw on the first suction sample. So we get a bit more information than simply the animal that we're collecting with every suction sample. Got
Yeah,
a little bit of swell on the ship today.
No, the containers are really solidly made, aren't they, and carefully designed so that the water's kept cold. They're sealed inside them, I think, so it's cold when it reaches the surface.
Yeah, really well insulated, I'd say. The bio boxes also, too, are made out of a really thick plastic material that's super well insulated. The temperature doesn't change much at all from the bottom of the seafloor to the surface, which is pretty amazing.
It is a lot like your vacuum at home, isn't it? Except you've got, was it 6 bin bags to fill. You know, when you've lost you vacuumed up, say something by mistake, like your wedding ring or something like that.
Oh, yeah?
And then you have to go through the bin bag to get it.
That's a pretty sturdy vacuum.
In case my wife is listening, I've never lost my wedding ring that way, darling. Just letting you know. It was
just an example.
Oh, yeah.
Hypothetical.
Yeah.
Seems like the gastropod is the size of the laser dot. It's super
So
small.
Where is it?
I think it's here.
Oh, it's still there.
So they're doing sort of trying to clean out the tube a wee bit before we slurp up this gastropod. Ooh, wow, and that was effective. Success.
Go for it, Chloe.
Yeah, hey folks, it's Chloe here, and I can vouch for what John has said. He has never lost his ring thus far on this cruise. And we can say a big shout out to people tuning in from home in Rarotonga. Hello, Nicola.
Hi, Nicola. Thanks for tuning in.
Thanks for tuning in. We hope you're finding this live quite a riveting watch.
And we hope you're enjoying your time alone.
Yeah, there you go. With the hermit crabs in the yard.
Thanks for the vote of support, guys.
All right.
A common question we get asked is why are nodules on top of the mud? Or are there more nodules underneath the ones on top of the mud? And so, firstly, there are some nodules buried beneath the ones on top, but not very many. The numbers vary between different places. In many places there are none. Some places it might be a quarter of what you see on the surface.
See it anymore. I can't see if that's the white object.
Underneath. Yeah, I think so.
It was, but it might have tumbled off with it can get disturbed, but if you can.
There, under the scoop. It's under this.
Oh, okay.
Are we going to hunt a gastropod, are we?
Yeah, if it didn't tumble off, it should be on top of the rock.
There you go.
Yay! I see it, I see it.
Oh, was just missed him.
Nice catch, nice
Yeah.
catch.
Yeah. It's like an Arabian sandstorm. We might name this gastropod Indiana Jones or something.
Or Lawrence of Arabia.
Lawrence, yeah. How about that, Lawrence?
We need X-ray vision, I think. That would help.
It'll clear. It'll clear fairly quickly. You can see it clearing. Yeah.
Where's Caitlin? She has X-ray vision.
That's right, Caitlin's.
There you go.
Well, it's been a good dive so far. We landed on a site with a relatively high density of nodules, irregular shape and sizes. And we're seeing a few different biological.
3 holothurians.
Yeah.
1
There you go.
acorn worm, a glass sponge, a few gastropods.
And about 50 trillion nodules.
Yeah, it's
Give or take.
A trillion.
fairly accurate.
Yeah.
Plus or minus.
And a handful of them being shark teeth.
Yeah, that's true.
All right, so now our pilots are about to suction sample a gastropod currently sitting on the nodule. This will be our second sample for this dive.
We think it's in the family Margaritidae. My mom's name is Margaret, so that's fitting.
Shout out Amanda's mom.
Oh. Yeah. Oh my God. Yeah, I think a scoop would have been. No, he's attached. He's not going anywhere.
You got him.
No.
Oh, dear.
Well, all I can say is it's riveting from where I'm sitting. Are we going to get this sample or not? Go. Take it.
I hope you got him.
Yeah.
So we're watching with bated breath at the suction sample chamber to see if the gastropod has been trapped. Yeah, that was good.
That's right. We're not going to need to take a nodule sample at this rate. Okay. Anyway, as I was talking about before, one question is why nodules are on the mud, and it's thought to be a combination of currents and actual biological activity. So as animals filter feed over the top of the nodules, they remove sediment from the tops and redeposit it in casts. Also the currents sweep in and around the nodules.
So we're going to say that that's a yes for gastropod.
Oh, what's that?
So that was chamber 2. Right on. Sounds good. Thanks, pilot. We'll record it as collected. Nice work, folks. Thank you, thank you. Perfect, Rima. Nice job. I hope that the commentary in the front row is being heard by the shore side because it's very animated and very good in terms of direction and information flow. I like it. There's a lot that goes into every single operation, including these collections. It's a well choreographed event. Nothing we do down here is simple.
All right. Whatever's left in the slurp is going to ride.
Yeah.
Cool. Nice work, front row. Yeah, pilot, thanks. We're going to hold off on that for a bit, see if we can. Let's see, another carnivorous sponge. And, one of the spiky ones. And then, yeah, we saw one earlier. We'll see if we can get another sea cucumber collection.
Ooh.
Oh, yeah. I think we might want to do a scoop then for this, and we can also get some nodule material and sediment. But we'll take a... Yeah, this is perfect. Fernanda, that's great. Yeah, this sea cucumber holothurian, it's in the Elpidiid family. We're going to take a collection of this, bring it back on deck, and get it identified. I think this is great, pilot. If we can make this happen, that'd be great. We'll do a scoop if possible. And get nodules along with it.
There we go. This is not, it's clearer than that, but anyway. What's the family?
Holothuridae.
It's in the family as well.
That's correct.
They have a
B3.
different level, don't they? It's a
3B.
species, yeah. There might be another one below it.
What's this little guy called, Chloe?
He seems like a Holothuridae, possibly a Peniagone vitrea. And then it'll be our third sample of the day. So we are about to do our third sample for this dive, and it's going to be a scoop. We're going to attempt to sample the Holothurian.
Using gravity, Levi?
Well, we'll get a lesson in
Very
how Holothurians
entertaining,
run.
Levi.
That's it. Lovely.
I don't, yeah.
Please don't
Gotta be
basketball.
very, very, very quiet. Sneak up on these Holothurians.
Yeah.
That's it. Slowly, slowly catch your Holothurian. And she goes.
Now?
No.
When they put it.
He was chilling.
Current depth is 5,358 meters.
Oh,
And we're gently collecting a sea cucumber from the seafloor, along with some nodules and sediment.
and just checking what the name of that arm is called. I think I saw it. Isn't it called the Predator?
Oh,
It's a multi-function ROV arm with haptic feedback, so the operator can actually feel if it pushes back on things. And it's designed for all kinds of industrial tasks, but often a very gentle touch is required. The holothurian is effectively neutrally buoyant. It's really, really light. A little bit like a balloon would be pushing that around. Thankfully the arm can move in all sorts of directions, so they're going to rotate it down. Okay, that's in the back of the arm. And you can see the bio boxes, or the bio boxes on the left-hand side there. They're specially insulated boxes, so they can take a sample and keep it isolated and cold for the return to the surface.
oh, okay.
There it goes.
Oh, there she goes.
Oh.
There it goes.
Oh no! Get in!
Okay, close it.
Wait, it might have went into the other.
Yeah.
It was good to see the sea cucumbers moving around. That was the activity that we were talking about earlier. That once they get all full of sediment, they can basically relieve their ballast and fly away. Move on to another spot to eat. But I could have sworn it fell into the other bio box.
Yeah, it might be at the back of the box, underneath the lid.
I don't know.
So are we pulling it a sample?
I was pause for a second, see if we can.
Oh, there it is.
Yep, it's there,
Oh, yeah, there it is.
and it'll be in port outer with some mud and
That's it.
part inner.
In the
Whoop
box.
whoop. Nice.
Good work.
Hey, good job.
Nice. Woo woo. Applause all around.
Remus should be happy. It's your favourite one.
Still on the lookout for my headless chicken, John.
Okay. We'll do our best to make you happy.
Thank you.
And some sediment went into port inner, just as a note. I would put that as a note, as a additional text, I guess. Yeah. Yeah.
Yes. Correct.
Roger.
That
3
was a
bio.
reading.
Yep. Nice work, team.
Yeah. I was literally on the edge of my seat.
The whole room was.
Yeah.
No one was breathing.
That was a great sampling up by our front row.
Okay.
That was our third biological sample of this dive. Thus far, we've collected a
Gastropod.
gastropod.
Anemone.
Yes, an anemone, that's right. And now a holothurian.
And incidentally, a few nodules.
Yes, alongside the anemone,
And
the gastropod.
And the gastropod, I think, came with a nodule too. Yep.
Yeah, great work, front row. Correct, that was a holothurian we just sampled. So that was our co-pilots. That's a great suggestion, Levi.
Looks like we've got a sediment sample on one side and a holothurian on the other.
So we've used our scoop and our suction sampling tools. We also have push cores, the claw, and I think that is all of it. So we're making full use of our sampling tools today. Kia ora once again to everyone tuning in. Happy 61st Independence to our beautiful country, the Cook Islands. We are here on the Okeanos Explorer in the west of the Cook Islands Exclusive Economic Zone. And we are currently on the 12th dive of this cruise. This is an area that's been explored. We've dived in only once before, and that was yesterday.
Yeah. You're at 5.3 kilometers depth. And it's a beautiful warm day up on surface, but it's a chilly 1 degree down where the ROV is working. We've got a a weather front coming through, I think maybe tonight or tomorrow. That's going to bring winds of up to 20 knots. But I think we're at 0.1 of a knot down here at the seabed. Just gives you an idea of just the contrast in the environment.
And then there's the other holothurian we saw.
Earlier.
Now only one of them is down there because we've sampled the second one.
You think he's pleased there's less competition for food now?
I think it's traumatized that a big square device came and took one of his fellow counterparts.
I'm not sure if holothurians quite have the brains to work that out, but yeah.
But yes, less competition for food down there.
All the samples we take are really important because we are going to get them DNA sequenced. There's a high chance that we're going to discover new species in this part of the sea because it's so far away from other sampling of this type. A lot of animals may look the same, certainly from a distance and even close up. They might have a lot of the same morphological features, but because evolution is constantly at work, you might find unique, or rather new species, I should say, when you go into new areas. All of the work we're doing is part of just collecting knowledge to understand the biodiversity in the seabed. We're really keen to learn more about more of the biodiversity in different parts of the seabed so that we can spatially manage the seabed as best as possible. The life found on the seabed is an important part of, a critical part really, of defining the habitat type or the ecosystem in that area. And scientists make, in fact, maps of the different habitat types so that if there is going to be any future development of the seabed, people are fully cognizant of what might be under threat or what might be impacted, I should say, from any proposed activities. So whilst it just looks like a fairly innocuous sample that was collected and potentially it will have academic interest, there's a really important reason why we're out here surveying and sampling the seabed to try and learn as much as we can to make the best decisions.
At the bottom of the screen. Pilots, can we just get a zoom in on that stalk in the bottom center of our screen? Yes. Is it potentially a bamboo coral?
It's one of these.
Oh, we've gone over him.
Coral, you think, Chloe?
Maybe a bamboo coral, but I could be wrong.
Maybe some of our viewers are actually wondering what these little black rocks on the seabed are. These are things called polymetallic nodules. They're a kind of chemical sedimentary rock. And they're basically nature's way of sequestering manganese and iron out of the water column. And they grow through precipitation. That's why they're kind of a ball shaped. They often have a small seed in the center, like a piece of sediment or a tooth or something like that. And over millions of years, layers of manganese and iron accumulate onto that seed and form a nodule. Nodules are found in all the world's oceans, but not in all parts of the oceans. So
if it's too tricky to find, pilots,
the Pacific, Indian Ocean
we can
all have some
continue on.
nodules. the nodules in the Cook Islands were actually first discovered in the 1960s, and there was follow-up research in the 70s and 80s and 90s, and more recently commercial explorers have joined researchers in the country. What we're on now is actually a scientific research cruise. There's no plans by NOAA to do any commercial development of these, but we want to try and understand the undisturbed state in the middle of this dust plume, but anyway, undisturbed state of the seabed at this time. So we'll move on now.
Yeah. Oh yeah.
Gastropod.
Do you want to direct them to keep them going?
Yeah, they are going.
I think they're going to zoom in on the glass sponge. Oh, let's see what it is.
It was a gastropod.
Oh, could be.
Yes, our pilots are just getting in place to zoom in to this interesting organism. We're having a bit of discussion in the back and what it could be from this far out. Adam and I thinking gastropod. John is thinking a glass sponge.
John's agreeing with you now.
Oh yeah, it's looking like a gastropod. Possibly.
Not sure.
Oh. Could it not be? I don't know, the shape of...
No, it looks like a gastropod to me. You can see the shape of the shell.
Yeah, the shape
the
of
top
the
is
shell.
on the right-hand side. Yep. That's great. Oh.
And we sampled a gastropod earlier in the dive. And then, John, what do you think of the shape of the nodules in view?
These are fairly common in the Cook Islands.
Yep.
There's actually one there on the left we're going past, and now that might be a tooth.
Yep.
The root of a tooth, but generally they're quite round.
Yep.
And semi-smooth. You can see the small dimples on the surface, which are covered in small bits of clay. There you go.
And a beautiful close-up of
you
this
can actually
gastropod.
you can see the feeding tentacle of the gastropod out there. You can see it moving in the water slightly.
Oh.
Yeah, you got it.
That's great, pilots. Thank you.
Yeah. In this type of red-brown clay, the nodules tend to sound proud of the surface, and they're believed to be informed primarily from what's called hydrogenetic forces. So the metals are coming out of the water that make them grow. But you can see they've all got a small coating of sediment on them, and some of them must come out of that too.
We've seen quite a few gastropods on this dive, and that one was a really interesting one. Like John mentioned, had its tentacle out. And that beautiful close-up we got. Now we're continuing to traverse this abyssal plain.
Just drive center screen of Sirius.
Roughly about 53.
He's leading the way for you back to 285.
6 meters at depth.
We're heading up the slope gently. It's a gently sloping slope. Sorry, we're heading up the slope, up the gentle slope, as gently as we can go. All right.
Yeah, well, actually, we'll just keep as consistent movement as we can uphill here to northwest. If that sounds good with you.
Yeah, that sounds good. Thank you. Yeah.
Sure.
Let's try and cover some ground because we're hoping to get a particular kind of animal called a carnivorous sponge. So
Okay.
we'll keep our eyes peeled for one of those. But anything else we come across that's different, we might have at least a zoom in, if not a sample.
Okay. Sounds good.
Yeah, we're continuing to see nodules on the floor here. For me personally, yesterday's dive was the most interesting thus far on this cruise because we saw no nodules.
You can see the decapod, yeah. Well, there. That's all right. We can keep going. We'll just, yeah.
Ooh, a shrimp.
Yeah.
Very nice floating shrimp.
I think
Well,
we'll be.
we can move on.
I think we'll be all right. Let's keep moving on. And yeah. Can they hear me?
There he goes.
Yeah.
All
Okay.
right, no worries. Yeah, look at that shrimp. A beautiful, brilliant red.
That'd be great. Thanks.
Oh, there he goes.
So, Rima, we saw quite a few of these shrimp when we did the baited lander, didn't we, during Wise?
Yeah, they were probably one of the first to arrive on site normally.
Yeah. And then they were followed, weren't they, by other types of...
Yeah, and then you'd find that the fish would start to arrive, or they'd kind of hang around for the amphipods to come in and sort of start eating them.
Yep. Yeah, if they didn't get the bait, they got the amphipods, didn't they? It was a little bit like some snacks while they were waiting for the bait to be opened up.
Yeah. So with your Z-bias so high, as soon as you go down at all, it's going to go down a lot more, a lot quicker. But you could just dust off and then push out ahead. Just get out of it. And I just discussed the plan with watch leads. We're going to try to keep some consistent moves in. So we can't afford to get off of the Sirio screen and then do like a long zoom or a long sample if we have consistent moves in. So we want to make sure we're not going too far off of there.
It's certainly a very consistent nodule field we're looking at. It's barely changed at all so far, both in terms of abundance, it's gone up and down a bit.
And distribution.
And size, yeah. Very homogeneous.
I think 48 was like level, and I would just go up and down as best you can. Yeah, I think you just need to fly down, command it down, and just keep it at 48. Yeah, and then just push out, like dust off, come down, push out, and then, yeah. And I just wouldn't mess too much with Z-bias. Just nice, small, incremental altitude changes.
Amanda, what are you hoping we'll see on this dive with the time we've got left here?
That's a great question, Chloe. I think I am really excited to see the seafloor and
Let's
be
try
able
to get your
to
mini zoos out ahead if you can.
examine a different location
blue
within
view.
the Cook Islands than we've been to before.
Yeah.
So this provides a lot of really great information on
thanks.
not only the terrain that we're seeing, the seafloor composition, but also the animals associated. So I think it's exploration in its truest form, and that's what I'm always excited about these dives. Yeah.
Nice.
First eyes on the seafloor.
What a great, well-rounded answer, Amanda.
Oh, thanks, Chloe.
I'm looking forward to hopefully seeing, like Rima said, a headless chicken monster.
I wouldn't mind seeing another one of those faceless Cascales, but I don't know if I can conjure that this time. That was fun.
I'm looking forward to seeing something very, very weird.
Weird,
I hope.
We'll keep our fingers crossed.
The weirder, the more intriguing.
And who's looking forward to doing all the filtering in the lab afterwards?
John is raising both hands up for that.
So yeah, our job isn't done when the dive is finished. The ROV returns to the ship with all the samples on board, and they've all got to go into the lab, and they have various different protocols for preparing different samples for different analyses, including DNA, fingerprinting, and carbon isotope work, and other chemical and mineralogical work. Some samples are sieved to try and find the smaller animals that we can't see with a camera, things called myofauna and macrofauna maybe. And so every part of every sample when we're back at the lab.
Yeah, I feel like so grateful to be able to work in this area. I rarely get a chance to work in abyssal environments, and using a vehicle like D2 and the ship to get out here is a lot. It takes a lot of work, takes a lot of people to get us here. So we do lean towards maximizing all the information we can from every single collection, as John was saying. It's a rarity to be able to get out here, and each minute on the seafloor is very valuable. And even the imagery is gathering essential information we need to better understand what's living here and the distribution of nodules and a better understanding of the overall environment here.
Yes, this is a good
and it's really a team
altitude,
effort, isn't it? This is
for surveying.
a ship with, I think, 51
you
people on board.
Turn your head and kind of like
and everyone's
mow
contributing.
the lawn,
The
like
steward,
back
the
and forth
cook,
if you want.
Everybody's
What you're
sitting on
doing
the
now is
stern.
also giving them a good,
and the vessel's
good volume
about 224 feet
at.
long. So I think it's about 80 meters or so in metric. And
there is something floating on the bottom.
And all of this is
I wouldn't
here
say
to
fast.
try and advance
I think
science. And a big part of what we're doing here,
it's
apart
whatever
from
you're
mapping
comfortable with.
and other water column profiling, is this ROV operations you see in front of you.
Yeah. I mean, we'll keep a consistent move in. You'll have to go a little bit quicker to get out in front of. So I think just to cover the most volume and see the most seafloor, like visually, if you just like nice and easy turn your head and then just kind of like snake back and forth, then we can get light on.
See all the nodules
a lot more
around
out
it,
there.
Every now and then, it's going past on the bottom now. There's a triangular piece, and those are usually
we're getting
teeth of some kind. Some of the bigger
good views
ones
here, so if there's something there, we'd see it.
megalodon teeth that an ancient megalodon lost while swimming in the overlying waters maybe over 1 million years ago, that fell to the seabed and has slowly been encrusted. So
because it took too long to see its dentist.
Yeah. They're messy eaters, aren't they, sharks?
Yeah, right.
Looks like that moves
about
done.
When we go to the schools, we used to show a picture of nodules with one megalodon tooth. And the child that could point out the tooth the quickest would win a nodule as a prize.
Oh, wow, I love that.
That was always pretty popular.
I want to come to that class.
Yeah.
Oh, looks like
that sounds great.
Sponges?
Yep.
No?
Maybe a holothurian.
It seems like something
and a
maybe floating in front of us as well. I don't know if we can set up for maybe this collection if we image it. But I know this has
And
been
then
a
a
winter.
shrimp.
Oh, jeez. Goodness. Okay, thanks. Let's see what we have here.
Oh, and what's this?
And shout out to TJ, who's tuning in from back in the office
Hey,
on
TJ.
the main island, Rarotonga. TJ was on the first leg of this cruise with us. Thanks for tuning in, TJ. We
Ooh.
miss your
Oh.
expert commentary. Ooh.
Interesting.
That's definitely floating, right?
TJ, what do you
Amanda?
Sort of
this
jelly,
is?
but I don't know which one. We saw, I think, a hydromedusa or something earlier on in the mission. Yes, pilot. If it's feasible to collect, yes. With the slurp. Or we can let it float by, which is also fine.
Yeah, I'm not sure if the suction sample will get down that far without trying to grab it. You could turn it on.
If we can get some good imagery, that would be great.
You might be able to get a good shot right here if you come back and turn right. Yeah.
The ROV is working hard, but that organism is working hard.
It's also who is going to win the race?
You got Tether.
It's in the cloud. That's all right. It's
That's
filtered.
okay.
Yeah.
That's okay.
It's a
Thanks,
filter
gang.
feeder,
That was
and I
good to
think
get a
it's,
take a look
yeah,
at it.
good buffet right now. Yeah.
Appreciate the imagery, and we'll take that back to the shore side for identification.
Thanks, pilots.
I'll
And
just
I
push
think in
out.
the background
Just push out
was
real quick.
potentially a holothurian to the right side.
Yeah, looks like it. Chloe, good spot. Another sea cucumber on the right. And we can... It looks like I'm also seeing piles of sediment, like piles of dirt, mounds
could
of dirt.
Be an acorn.
Something is creating that, either living on the surface or living subsurface, below the sediments or below the nodules. What's over on the left, pilot? I'm going to make you focus on the thing that's swimming. Please.
Top left of the screen.
Yeah.
Yeah. Chilling in the distance.
Yep. For now.
Oh.
Yeah, looks
What
like
is
it.
that?
I don't know.
That dark thing
That
looks
dark
interesting.
thing in the background.
That could be a headless chicken monster.
But yeah, the one in the center may swim away from us, so if we could capture that, that would be great.
Correct.
Right on the lasers.
Between the lasers right now.
Fish?
Ooh,
Oh, yeah.
what kind?
Gotta get a good look at its head.
What do you think it is? Yeah.
Yeah.
It's pretty big though.
Yeah, that is a big blob.
Well, looks like we're gonna zoom in on this
Oh.
fish right here,
Kind of look like it.
which isn't showing us its head as usual. Probably some kind of cusk eel.
Oh.
Looks a little rough around the hinges.
Does it have eyes or no eyes?
Yeah, it does have eyes. It's not what I was looking for. But I'm happy to see a fish. Always a treat.
Amanda would have been happier if it had no eyes.
Yeah, I should stop like having expectations. No, this fish looks like it's in good shape. Some kind of cusk eel.
Yeah, he's chilling.
He's frowning.
This is chill face.
Looks like the same species we saw yesterday, maybe,
Yeah.
or same form anyway.
Yeah, it's got some ectoparasites on it as well, but not quite as colonized.
Yeah,
And
that's
then that brown, the purple object
Let's look at
over
that.
there.
Let's look at that.
There's
please,
a holothurian,
pilot.
yeah, you can see the track. And a nice big one at that.
Yeah,
That's got to be, what, 20 centimeters long?
Yeah, it's
Maybe
gotta
more.
be.
good spot. You can see the as John was remarking, the
Sure.
track that the holothurian leaves behind while it's feeding on the sediment, on those deposits.
Watchley, is this something you want a nice zoom of, or?
A zoom would be great.
Great.
Yeah.
I think it's good to touch base too, because if they don't need that zoom, then we can keep going.
Yeah, we just haven't seen this on the dive so far, so it would be good. Something different.
Yeah, sure.
Is that a headless chicken? Monster?
Well, it's the right colour. I'm not sure if it's the right shape, but
Very cool.
You're still creeping forward.
Is
I
that
think
an
this might be a
amphipod
different
on it?
Yeah, nice.
family,
Ooh, look at that.
but I could be wrong.
It's your lucky
And
day, bud.
striking
That's really
color.
beautiful.
You're still creeping a little.
shall we try for a sampling that one? That'll be a good one for the museum.
Oh.
2 thumbs up for me.
Do we have time for a sample, pilot? And there's a little polychaete worm hanging out on top, going for a joyride on
You're
the
still
sea
coming
cucumber.
forward. You need to come back a little.
A little white, sort of iridescent worm.
Yeah, there you go. Nice.
It's also likely feeding on food on there.
Yeah, this one looks like it has a head.
I'm wondering if it might be a different family than the headless chicken
It looks
monster.
different,
Looks
doesn't
different.
it?
I think it might be of this. It might be a synallactid, but I'll look it up.
And you can see the small worm swimming past it on the left there. Yeah.
Oh, little worm.
A 2-for-1 deal. I love it. Wow, look at that striking.
You could probably turn
dark
the
red.
facing
yeah.
if you want, Fernando.
Really pretty
We're not moving.
contrast. Yeah, it's a synallactid, which is the family for the sea cucumber. It might be Galatheathuria or something related. Really pretty.
Yeah, if any of our onshore scientists are tuning in, what do they think this is? Oh, definitely a different family.
Yes, we can collect. This would be great. Yep. We would need to scoop it.
Scoop.
Scoop it. Scoop it.
Scoop City. And unfortunately, it sounds like there is a swimmer in the port bio box. Yeah, which I think means we just don't try port at all. Yeah.
Roger.
Yes.
Yep, 4B.
All right, our pilots are getting into position to sample this organism.
So one thing you could throw in just to
Potentially a
eliminate another
synallactid
variable is auto heading. If you
Galatheathuria.
wanted to.
Maybe.
You don't
Maybe.
have to, but
Maybe,
it's
maybe.
just something to think about.
I wonder
like
if it's in-
conditions were more.
What do you guys have in APSMA? You have that pulled up. Wonder what they saw.
No, it shouldn't be working at all right now. It's more if you hit the ground and move the vehicle, it would like steady up for you and not just keep rotating. Yeah, but it's up to you. Right now, it's not necessary, but just something to think about if conditions were a little bit higher. I think. Yeah. So you are pretty tight to it. So you'll have to drawer out. I think the drawer will clear, right, Fernando? Over it, right? Then you grab the scoop and then do your thing.
That's a great color.
You sometimes find similar looking ones like this in the lagoon, but
Oh
they're black.
yes,
Yeah.
they're black. Sometimes the ones that look like this are like dark green.
Did you say green?
Yeah, I've seen dark green, like a really deep green color.
Oh wow.
Do they hang
It-
out by the like coral heads, or
Yeah,
where
yeah.
do they?
By the
Oh yeah.
coral heads.
Yeah, by the coral
ones
heads
that look
usually.
like this one.
Okay, that's neat. And you don't pick. These are the ones you don't pick up.
You
Yeah,
know,
definitely.
Don't pick.
definitely
We
not.
don't play with those.
We don't play.
Have
with
you
these
ever eaten sea cucumber?
Yes, so we
Yes.
eat them when they're dark brown,
Oh, okay.
but only the insides.
Only the insides. Hmm.
Let me know if you need the meat allowed.
What does it taste like?
People.
Salty noodles.
Oh,
Yeah.
interesting.
Tastes like
makes-
the ocean.
So it's kind of a. Is it chewy or gummy?
Gummy.
Yeah, is it more like-
Yeah, a
okay.
little gummy. Kind of like an oyster where you just sort of want to swallow.
Yeah.
Don't really bite it
Yum!
that much.
You're really making it sound delicious.
You really only eat this while you're in the water.
Yeah.
Oh, okay.
Yeah,
And is it only done at a certain time of the year?
I think so, yeah.
Under the raui? You want to explain how that works a bit?
Oh, so in some areas or lagoons around the island, we have protective measures over them. So there might be a season where we don't allow full collection, and we usually call that a raui.
Oh.
And
So
then
no take.
it allows for the resource to grow again, and then the island may open it up for a few months before closing it again.
Oh, okay. Interesting.
I just claw it.
All right, we're gonna use the scoop to attempt to sample this sea cucumber.
I think we're going to need a bigger scoop.
Watch it if you need to. Yeah, just. Okay. If you're indexed out, then you have it locked. Because otherwise, the jaws would have opened. So you should have. Well, yeah. Yeah, we'll give it a shot. Well, actually, not sure if this is going to be big enough, but we'll try.
Okay.
No worries, front row.
As long as you get the center of gravity in there, you'll probably be all right.
All right, looks like it's roughly the same size as the scoop.
I think it's bigger.
That's 30 centimeters at least in length. That's all right. These boys have got a technique.
Yeah. Yeah. I think you can't come on it like halfway through because the scoop is not flat. So. Yeah, I think that's the right idea.
So Noah has this benthic animal guide with images from previous NOAA expeditions and a sea cucumber that resembles the one that we're trying to collect today in the family Synallactidae was collected or imaged at a depth of 3,868 meters. So
No, that's all right. making
It's a
room.
deep dwelling species, and it was in the Mariana Trough
Oh,
back in 2016.
Oh, here they go.
Yeah.
They're attempting to
Ooh.
scoop it.
yeah, it's full of sediment and nodules.
Please don't fly away.
Yeah, and I would keep it tilted like that. I think. Yeah, there you go.
He's just bouncing on there.
Yeah.
It's like holding a tray of
And its
jellies
little
real,
cradle.
real slow.
with no lid on it.
Little D2 cradle.
Yeah, let's. If you need, you want me to turn people off if you need it. Okay.
You could probably put that in the geo box anyway.
Unfortunately.
Oh gosh. Catch.
Catch him.
I think you're clear of the box. I would try to extend the drawer, make that available. I think that's out. Yeah.
Oh, he's going to roll off. It's rolling off.
You got him. Catch him.
No. You gotta get the center.
Oh, at least we can see the gastropods.
He moved on you.
It's on the tray. Off the tray.
It's amazing that polychaete stayed along for the ride, little rodeo ride there.
Yeah. I can. Do you want me to fly it back?
Just pull back.
Yeah. Yeah, bring the arm up a little.
I don't think it had this, like,
I was gonna take him.
I don't think it had this little skirt around it, or not.
Yeah, just keep doing that. Nice and easy. You got some tether on Sirius here. Can you come a little port, Fernando? Just keep it in view. Yeah, nice. Thank you. Watchley, you want to give us one more shot?
Sure, let's give it a try if you can.
Yeah, that was a yeah.
Yeah, it looks like the scale worm is still hanging out on this sea cucumber that we're trying to collect today.
It's interesting. I thought it would have emptied itself and flown and swum away, but it's playing possum or something like that.
Yeah, I was going to ask if it would fit in the section or not, honestly, but if it's too big. That was a suggestion. Only if it's possible to, if it will fit in the tube. I'm not sure if I'm the peanut gallery, if that's also elsewhere.
Well, that's definitely gonna. Something will emerge from the suction tube, yes.
Yeah, thanks for giving it a whirl, pilot.
Yeah, we're in position and
Hey, video. Hi, video. It's the same sample. Thanks, video. Hey, video, are you able to get the pilot GUI in the camera 3 quad? I think the shoreside's been requesting a way to see depth. Oh, I see, I see. It switches out. Okay, I didn't realize it was switching. The jaws have coral cutters on them, I believe. So it would be difficult to grab it with the jaws, I think, without maybe causing some damage. Oh. Jar 3 should be empty. Yeah, 2B. Sample 2B is in jar number 2.
Onshore is saying this one is so juicy the suction actually might work.
Yeah.
That's it.
Maybe we'll get the little scale worm as well.
Ooh, yes.
We'll see. Well, where are we, Rima? What are we doing?
We're currently down on the abyssal plains doing dive number 12. We're in the northwestern part of the Okeanos. And right now we're attempting to collect this juicy sea cucumber that you see on the screen.
I know you wanted to see some sea cucumbers today.
I did.
Your wish has been granted.
Tomorrow we will see a whale fall.
There we go. Stranger things have happened.
All right, now our pilots are attempting to sample this holothurian with a suction.
Oh, they're doing a great job. They're shuffling the ROV around, they're shuffling the hose around, they're shuffling the trays around just to try and get it all into position. Because it is such a large specimen, it's a little bit trickier than normal.
Well, he might block the suction tube, guys.
Oh, that we're hoping not. It seems like the nodule composition's been really similar throughout the dive so far. There's some small and larger size nodules and more irregular sizes. What's that a function of, Chloe or John?
I think pretty good. Yeah, I'll just get a little closer.
So
you're going to get sediment, so don't worry about doing that.
nodules are very slow growing and they reflect a lot of the conditions that's existed around them for literally millions of years. And this particular part of the seabed we've landed on is a very smooth, continuous slope.
Go for
It's likely that there's been very consistent currents over this area. And I'd imagine that's really what's helped form it. There we go. That didn't take much force.
Oh,
there it is. It's in the sample tube. You guys can't see that, but we have a camera on the sample tube. And it's somewhat full of holothurian, so the slurp was the go after all.
Nice call, shoreside.
And we successfully collected
various input from the ship. Thanks so much.
We're going to preserve that when it comes on deck, and hopefully it'll. Sometimes they change form of it because we use ethanol, but if it's in good condition, we're going to see if we can put that one in the National Museum of the Cook Islands.
Hooray!
Yeah. Okay.
Nice work, team.
Great work, front row.
Shoreside is dutifully impressed as well.
Hold on that, I'm not sure if the hose will let you, but yeah, we can try.
And I guess whenever Nav has a moment, maybe we could estimate the distance covered so far.
Yeah, give me a second here.
Yeah, sure, no worries.
And time remaining, please.
Understood.
So Amanda, you're hoping to also find another carnivorous sponge to sample, is that right?
That's correct.
Do you want to tell us a little bit about what carnivorous sponges are while we're watching this come back together again?
Sure. The carnivorous sponges, we find a variety of different kinds on the seafloor. Different groups are represented. Some of the ones that we've been seeing on the mission so far on the cruise include ones that have these really amazing basically tops to them. They have spines that come out from a central disc area, and those spines are covered with barbs or hooks, and those are adapted to capture food that it's floating past. And then the sponge themselves, they actually envelop the prey that they trap with their hooks in sponge tissue and digest it that way. So there's a well adapted to living in the deep sea, feeding in really food poor environments, capturing food opportunistically.
I was just trying to grab the ball.
And make a living.
Just be
that
real
way.
mindful you're in
Pretty
tight.
amazing. And sometimes we see animals that are living associated with the sponges. The sponges aren't eating them. They're not using them as prey, including anemones that we find attached to the
So you indexed out while you had a
itself,
command in,
and then
so that's
also
why
on
the jaws
the
are closing.
axis or stalk of the sponge. So not only do they trap food, but they also provide a home to other other animals.
There are other kinds of deep sea sponges, including glass sponges and lollipop sponges and all sorts of forms. But carnivorous sponges are a distinct set of forms, and is that because of the way they catch their prey? Because they must all be carnivorous in a way, living so deep in the ocean.
Well, there are deep sea sponges like the hexactinellids that we've been seeing, the glass sponges that
I would try
are
to
filter
grab it.
feeders,
I wouldn't try to
and
touch
they
it.
basically actively pump water through their bodies and trap food that way, and that can include living and non-living organic matter.
Okay.
Yeah. So detritus. They also have microbes associated with them that help them with their nutrition.
Yeah, I think.
So a lot of really complexity in what appears to be a relatively simple organism.
Yeah.
And you need to go down more. Yeah.
And do we know about the evolution of sponges per se? Did sponges in the deep sea come from sponges near the surface, or did sponges at the surface come from sponges in the deep sea? Do we have an idea of which way they vectored?
I know very little about the sponge evolutionary history, but would love for our shoreside scientists to chime in, or if anyone here has any knowledge. There's a lot of complexity in sponges that we find in tropical coral reef environments, and work that's been done in coral-related sponge habitats. The sponges actually play a huge role in cycling of organic matter and pumping carbon and energy through the system. They're an important filter and also habitat. So there's a lot of parallels in terms of sponges that we see in shallow water environments that we see in the deep sea as well.
Yeah, come out with the don't come anymore on your azimuth. Look at Mini Zeus, you're in real tight. So
now
I
I'm
think
really on the lookout for sponges because we've been talking about them, so I hope we see more.
Is that like when you're talking about something, about food and you get hungry for dinner?
Yeah, that's about right. I think that's it.
But for the marine biologist.
Right.
Yeah.
Waiting.
Oh, my wife's a bit like that when you talk about diamonds.
Oh, dear. Oh, dear.
Thankfully, I'm not the same way.
Hey, Nav. Wow, 210 meters. Rock on. Okay, great. Wonder how much ground we covered yesterday. It was over 200 meters. Yeah. Yeah, that's fine, Nav. Thanks. I don't want to distract. Yeah. Easy enough for us to figure out at some point. Good job, front row. Nice collection.
There we go.
It works.
Our pilots are just slowly and carefully putting all of their sampling tools away.
Has anyone besides Chloe and John found a duck yet?
You found a duck.
Look,
I was so jealous.
it's like a Viking duck.
Where was it?
That was my coffee machine, yeah.
Oh,
yeah.
wow.
You're going to have to explain to all these people what these ducks are about.
That's good.
Go
It's
on.
a good story.
Yeah.
Someone on the ship has skillfully hid 60 ducks overnight. And for every one duck you find, it
sorry,
equates
just to make it clear, these are not real ducks.
Rubber ducks.
Rubber ducks,
Yes.
okay, just checking.
every one duck you find equates to $1 at $1 at the ship store. And currently, between Rima and I, we have 2.
I'm very jealous.
Or
Also,
Chloe,
thank
John,
you,
and
pilots.
I for 2.
That's all right. I think the person who's jealous in the corner has actually found 4. Is that right, Amanda?
I found 0.
We
Oh.
I found
thought
0.
it was Amanda, but it's night
the other
shift
Amanda.
Oh.
There's more than one Amanda on board.
Okay. I was confused at first when I heard it at breakfast. They were talking about duck, and I thought, well, is it duck for breakfast? It was a bit odd.
Delicious.
But, yeah.
Oh. Now you
No,
can cash
you're
in
on.
your
You can't
ducks.
go. You have to wait.
Maybe
the
tomorrow.
store opens every day for about 15 minutes. They've opened it right now. But Chloe's on watch, so she's not allowed to go and cash in her duck.
No.
Nav, you said there's 2 more hours left on bottom?
-1.5.
Ow. I know. I have to try once a cruise. Push the envelope.
All right, everybody, we've started again on our traverse up the slope here in the Western Cook Islands. We had a very protracted sampling procedure, but we've got a fantastic large holothurian. And now we're on the final hour of our traverse, and we've got one particular animal that we really would like to sample, and that's a carnivorous sponge. So we'll just have to wait and see what nature's put in our path. But imagine us a little bit like big game hunters, but scientific ones, and imagine Amanda.
I think we can sneak up on the sponge.
You reckon?
I reckon it won't walk away.
You don't have a blunderbuss, but you've got a Slurpee, and you're going to see if you can hunt down that
Got a few.
carnivorous sponge, yeah. And if a lot of you think it's a bit like a triffid or something like that out of science fiction, carnivorous sponges are normally a few centimeters to tens of centimeters tall, and yeah, they're pretty dangerous if you're a small plankton or something like that, but I don't think they're going to sneak up on us, are they?
No, I think we're safe.
Yeah.
What else do we want to try to encounter on this dive? I think before we leave bottom, we want to do some water and some push cores.
So we are actually approaching the base of a small knoll. I don't know how close we're going to get. If we get to the end of the 500 meters, we might see a small outcrop of rock. But it's just a question of how far we get and the time we've got left.
Oh yeah, crinoids are on the wish list from our shoreside scientists.
Okay.
Hopefully get to see some sea lilies.
I don't have them.
What's that?
Just looking for the charger.
Yeah, sure. Let's do it. You guys good with another 30 meters? Okay. If we see something that we might want to sample or do a more imagery of, we can always come to an easy stop, but we want to do that pretty quickly if we determine that.
So whilst we have all these aspirations to sample more life, the biomass at these depths is extremely low. This is in many ways the equivalent of a desert, an underwater desert here at the base of the sea. And in fact, the biomass in some of these environments are less than what you'd find in the middle of the Gobi Desert or the Sahara Desert. But deserts have life, and that life is really important to understand. And it's the same here.
Uh-huh. 4-1, yeah, yep. Yeah, you could like change the set point maybe, see what that does. Yeah, I guess there's not a ton of current. Well, he might pull on the tether a little bit. I don't know.
Number.
Did you see something?
I think it was just a holothurian.
Okay.
What is this?
Cool.
Gastropod.
Yeah.
Heading.
Sponge.
You can actually see the slope, eh? We're heading due north now with the ROV.
Is that a sponge, Amanda?
Straight ahead?
Below the lasers.
Yeah, looks like it could be.
Not the sponge you're looking for.
Yeah, it is one. Yep, it is a carnivorous sponge.
Yep.
Do you want it?
If we can, I just don't know what we could put it in. But thank you.
We still have the bio boxes, don't we, on starboard?
That's correct. We could zoom in on this carnivorous sponge. I think it's a clatterized. It's on the small side.
You still got auto head in, just so you know. You're good. Yeah. How would you recommend sampling this spot lead? Coral cutter?
Yeah, it's attached to the nodule. I think cutters may work best if you can grip well.
If we could what?
If you can grip it.
Oh, the nodule? Or
Grip the base of the axis of the sponge. Yeah.
Ah.
Or get close to the base. Is there like rubber tubing on the other side or something?
Yep.
Okay, that's cool.
Yeah, so we'd put coral cutters down as close to the ground as we could. Right?
Yep.
Is that the idea?
If possible.
Okay.
Depending on your reach. Yep.
Yeah. You're going to have to come in. Yeah. But that was a good, just so you don't dust it up, it's good for imagery, and then, because they want that too, so. Yes.
Yes, video.
Lip balm.
Copilot, what'd you determine from that test? It was pretty good, right? Oh, cool. Okay. Cool.
For the nodules. Yeah, if we've got time, let's get this sorted, and yeah, we'll leave it at the end. We've got something at least already.
It's pretty tight. I think you're good though, as long as you can get down. Well, yeah. Let's see. Let's get eyes on it.
I was
On
wondering what
mini
the
zoos.
abyssal guide has for the characterize. And the benthic animal guide, we've got a few options.
I think the idea is to. I think they were saying coral cutter, yeah.
But I'll definitely leave the final ID to the experts on shore. So.
Right.
Taking a pretty big roll here. I'll bring a serious view.
This one
I got
called
it up. Never mind.
Exonoderma, which could be similar.
Don't forget about your fogs too, if you want to try for more light. Oh, nice. Yeah, I think that's a good plan. Get your arm in the right horizontal, and then bring shoulder down, just like you're doing.
So what other additional collections were you guys hoping to get from the dive?
Yeah.
What other additional collections were you hoping to get from the dive?
Possibly.
Yeah, I would center it up.
Scuba nodules. Okay. Okay.
I'm thinking like something more interesting, like
And I would like
jellyfish,
like bring the jaws in from the side.
Excuse me, nodules are interesting.
Yeah, exactly, exactly.
Oh, there you go.
So one of the issues is you've
got more regular forms here. I don't know if you realise compared to some of the other ones.
You see the wrist. So your wrist is going to touch bottom before you get down there.
Variety of
So
shapes,
you do,
sizes.
Yeah, so you come in at an angle if you want. You could try that. I think there's enough clearance. Yeah, or you could technically rotate the jaw and then come around on the other side. You could do that as well.
Would you, after they collect the sponge, would you guys want to try and do a scoop into the rock
Well,
box?
you'd have to turn your whole arm
If we
around,
got
so
space,
it was still on bottom.
Yeah, rock box is plenty of space.
Yeah.
But I don't know how that.
Oh, sorry, of course, yes.
That'd be.
Well, let's do it because we've got the irregular forms as well.
Yeah.
Yep.
Is this okay in terms of a sample?
That we don't have to move.
There you go.
Yeah. Maybe they could pick up one of the larger ones.
And I would
within reach.
Keep an eye on midi Zeus because you're out there really far.
Yeah, we'll do the sponge first, and then we'll get a scoop.
And don't worry about the sediment that's in there.
And you're going to touch here. You're going to touch if you keep going that way. So I think I would shoulder azimuth in. Yeah, there you go. I think you need to elbow down, but you might be too close. Yeah.
And in fact, if you want to go for 2 scoops, that
Yeah.
might be a tooth on the
I
right
think the only way you're going to really get it is if... Well, you might just be too close to it. But you could keep doing what you're doing and maybe try to, like, you can come at an angle from, like, this way. Yeah, there you go. Yeah. Nice. I think that's a good setup. Yeah, just nice and easy. Use both cameras. Yeah. Yeah, so now's a good time to, I think, check in with Watch Lead and just make sure Amanda knows the plan. Like, you're going to try to cut it as close to bottom as you can. The jaws are still kind of tall, so you might come down on the end of the sponge, and you could just see if that's acceptable. Yeah, I don't know. Watch Lead, are you? We'll try to cut as low as we can. Is that okay?
Yep, that
Okay.
works. Thank you.
Yeah. Well, she said that's good. So I think as long as we're as low as possible and in the jaws, I think you can go for it. You know, you might have a little bit more elevation to go down. I think you can go down more. Yeah, there you go. Nice. See where you're hitting it? I would hold on that and go down more. You need to get the blades all the way to the ground. Yeah, down, 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 even more. Yep. Yeah.
Can see a little amphipod hanging out on
Yeah,
the
and even if you need to angle,
base of the
you know,
sponge.
I think that's fine too. Yeah, nice. Yeah, and that's fine though, because you're gonna need to go as far down as you possibly can. Yeah. Nice. Yeah, I would go for it. Yeah.
Good.
And don't forget the jaw lock. Yep. Great.
Oh my gosh.
Someone's excited about getting bingo.
No, I think someone's sad that someone else got bingo.
Oh, yeah, I understand that.
It's all right. I got eyes on. You're good. deeper. Yeah, but definitely release slowly because you don't want to. Yeah. And you can see how high you are in the port manip. Yeah, keep going down, keep going down. And outboard more if you can. Okay, I think that might fall down in there if you want to slowly open. Yeah, there you go, nice. The jaw lock. Yeah, there you go, nice. It's still in your fingers, so yeah. I would feather your fingers in, and then, yeah, come straight up from there. Come straight up from there. Yeah, nice. Nice work.
All right, sample collected.
Starboard inboard.
That's correct.
Thank you. Nice job, folks. SI, yeah. And then for this one, oh, sorry. Go ahead. Yep. Dominant, yeah, good. Good. So before we take off front row, we'd like to do at least one, if not two, scoops into the rock box.
Is this oval long one, John?
You want that one? Yeah, we can go for that oval, that long oval one right in the center, behind the disturbed area. The nodules here are a bit more unusual, so let's see if we get something larger.
That's a science question, I think.
Yep, this is fine.
Yeah,
thank
we're
you.
fine with the scoop. It's going to go in the rock box, and I think the team was hoping for the, I guess, the nodule that's sort of long by the disturbed sediment, bottom center. Is that right, folks?
Yep.
Okay. Right on. Yeah, or the, yep.
Can we drop the camera, please, as well?
Tilt it down a little bit. Yeah.
Now, if you look on the right-hand side, after you picked up that oval nodule, there's another irregular one on the left, and it looks like it might even be a tooth on the right. I don't know if you can access those, or are they too out of the way? So if you're looking at this, I think it's Zeus camera, just on the scoop, just on the edge of the scoop there, you can see there's like a triangular nodule. You see that? It's quite a large one, yeah. Right. And then on the right-hand side of the field of view, just near the skid or the edge of the box there, it might be a tooth. So the order I'd go for these is, first I'd go for that oval one that's right in front of you. I think just take a scoop and see if you can include that in there. And then secondly, the one on the left, just near the scoop where it is now, and then we'll just see how long that takes. And if we've got lots of time, we might go for that. I think there might be a tooth on the right, but they can all go in together. They don't need to be kept separate. Just in one of the rock boxes is fine, thank you. Oh, and you don't need to clean the scoop. Just use it as it is.
Yeah, we're watching you.
Are we doing this geosample?
We are doing a geosample video. Correct.
Geosample? Yeah? Oh, yeah.
Roger, Nav, that would be sample 6.
What's the geosample?
It would be 6G.
Say the geosample again.
6, yeah. So Chloe, earlier on in the cruise, we had a nodule collection, and in the lab, we got to open it up, right? Remember?
Yeah.
What did it look like on the inside?
It was a mixture of nodule, crust, and sediment, and that's typical of the area it was found on, which was on a seamount slope.
So it had kind of a layer of crust, sort of outer coating, and then inside looked like compacted mud, right?
Yep.
It was just brown, dark brown in color.
Yeah,
Yeah, we get those in the Cook Islands, and we're not 100% sure what that internal mud actually is. If it's just a piece of the original clay on the seabed, or if it was a volcanic fragment of some kind, like a pumice that might have flattened, or we don't know. Okay. You can go for either one of those two samples, whichever's easiest for you. If you want to go for the one on the left first, go right ahead.
What was that? A jelly?
Looked like it might have been a tunicate. Or no, it was a ctenophore, sorry.
Oh, ctenophore.
Yeah. Floating by. Different kind of comb jelly. Wasn't like the one we saw the other day.
Yeah.
Ooh, turning all the lights off.
Right ahead. Yep, that oval looking one looks a bit like what's that sweet you Americans eat? It's like a
A Tootsie Roll.
what's it called? Tea roll? No,
A Tootsie Roll.
is that what it looks like? Okay, the one that looks like a Tootsie Roll.
I don't know. Is that
A Twinkie, isn't that what it looks like? A Twinkie?
Maybe the shape, but
Yeah.
not the color.
Oh well, maybe one that you left in the oven a bit long.
Yeah, there you go. A burnt Twinkie. Yeah, it looks like we have a little less than an hour left in the dive. And we've been traversing an area, relatively dense nodules, different shapes and sizes, really in contrast to our dive yesterday on the abyssal plain with soft sediment composition. We've got a few collections so far, including a really enormous sea cucumber. And we also collected type of carnivorous sponge and a different type of holothurian and a gastropod. And before we leave bottom, we'll get a few scoops, at least one here, and then possibly some push cores and water samples as well. So we can get a better understanding of the environment within this region of the Cook Islands.
All right. Put it in the bio box. I think we've got something anyway.
We'll
The geobox. The geobox. Sorry, my apologies.
Yeah, rock box, whichever one's most accessible, pilot. What was the approximate depth for our dive yesterday? About 5,400?
I think so.
Okay. Thanks.
I think I can give you a depth. Stand by. I don't have the recent multi-beam, but I'm guessing 5,500.
Okay, thanks, John.
Oh.
Did you guys see if it went in the forward or aft? Okay, we'll check both. Okay. John, do you want to continue scooping or do you want to move on?
I think we're good with here. Let's move on to the... Let's go a bit further while we've got some time, and then we can take a final sample, maybe when we do the fish course.
Okay, sounds good. Yeah, we're good to move on whenever you guys secure the scoop. Thank you. Sorry. This is 6G. And you had it, oh, 06. Yep. And then characteristic assay to put on geology. And then nodule collection, yeah. Perfecto. Nice job. Really? Do you have socks on? This is a cool one. Well, yeah, it's one of them. Okay. This part is. on this. I'm not sure which the anemone one is this. It's basically, this is the same group. They just don't have an ID on it yet. So, but yeah, this is the sponge part, and then the anemone's hanging out. You can see like the tentacles. She's not there. We'll have to figure out what our depths will be for our water samples at some point. I think so.
Yep.
Yeah. Might as well do the same ones. Yeah. 45 minutes. Yep. Yep. Seems like a thin veneer of nodules on top of sediment. I wonder how deep that sediment goes.
Yeah, it's a good question. The seabed here is quite old. It's the abyssal plains are believed to be around 120 million years old.
That is pretty old.
And so for 120 million years, the sediment's been accumulating. Now, some of it might have been moved over time by some currents and things like that, but certainly it might be possible to take a core and date different layers of the sediment. The biotubation mixes up things like fossils and so forth, and this particular kind of sediment doesn't tend to consolidate into a rock. So, for example, if you're at shallower depths above what's called the calcite compensation depth, the calcite lysocline, you can form a rock called chalk, and that chalk, once it's basically cemented together, can't be moved by burrowing organisms and things like that. So they can use microfossils to date where you are in the chalk. But when you're in this siliceous material, sometimes a bit trickier.
Yeah, they're just having a watch change out front. Front rows, shifting out some people, personnel. We hear you loud and clear.
We have about, let me re-update it, but we have less than an hour.
Less than an hour is what? Is that 55 minutes there, Amanda?
15.
45.
I think it's 59.999.
We have 45 minutes.
Gotcha.
Oh my
gotcha
gosh.
now. 45 minutes.
That's less than half an hour.
45 minutes.
45 minutes is more than a half an hour.
Okay, 45 is a great compromise.
I was like, if I did my math right.
Yep, you're good, Jacob. You're also fresh.
Yeah, I've been. Well, I was here this morning.
Yeah. You had a wee bit of a break.
Oh, yeah, come on.
Everyone's joining in the crowd and the fun today.
Yep.
We can keep strolling along.
We're going to be moving 277, and I'll do 30 meters.
What happened?
Bridge, Nav. Hey, can we get a ship move range 30 meter, bearing 277, speed 0.2 knots?
But yeah, pilot, we're looking to at some point get
That's
another
correct, thank you.
scoop and a couple push cores and a water sample before we leave bottom. So a little bit of extra things to do before we head up. Water.
With the right to reserve a biosample of something especially charismatic, perhaps. What was the other thing you mentioned was sought after?
And then you have
we were looking for a
in the port outer
crinoid.
A crinoid,
box,
okay.
just heads up.
And we got the sponge, and we got the gastropod.
Don't want him getting away.
And
You can vacuum him
we
up.
got
That'd
2
be
sea
cool.
cucumbers today, so that's really amazing.
Yeah.
You want to vacuum it?
You guys need to come off of, you're both on mute.
No, we don't
Oh,
want
it's a
that.
shrimp.
No, we don't want the shrimp.
It's a shrimp.
We had some of those for lunch.
I just wanted to see him vacuum something.
Not me.
Everyone was so disappointed. Poor little guy.
I think he's probably more relieved.
Yeah, really.
Yeah.
Please don't, you don't see me, I'm invisible.
What is this?
Charlie horse, that's what we call it. We used to have red lasers. Did those go away with a different camera? Or were they? Oh, okay. Okay,
This
thanks.
green one's really good.
Yeah, I like the green. I like the red. The red I can find easier, but the green are nice too. So I don't know. Oh yeah. People make sound effects. Pew, pew, pew. Yeah.
Yeah.
What's that in the distance?
In imagery, we sometimes lose the red ones against the mud. Whereas I think the green ones, you can always see them, yeah. So I'm definitely more a fan of the green.
Oh, you like the green? Whoa.
I just did 2 of
That's
each, why not?
crazy.
Disco ball, yeah.
There you go.
I'm guessing a glass sponge.
Oh, in the middle? Maybe.
Oh.
Definitely seeing piles of sediment, which may be a good indicator of bioturbation, of animals living in the sediment, moving mud around. Looks like, yeah. Looks like there might be a sponge. Good call, Chloe.
I'm guessing a glass sponge.
Yeah.
TJ, if you're tuning in.
There's a shrimp that still wants to be imaged.
Oh.
Oh, what's that?
The fish. Is it a fish? A shrimp?
Oh. This is a teeny tiny sponge.
Oh yeah, look at that. Very small.
Almost looks like a tulip.
Yeah.
Type of glass sponge. You can see all the pore spaces within the top. That's great, front row. Thanks so much. Thank you.
A hextinolid.
Yep.
The species of glass sponge, the scientific name.
Nice. Still seeing a fair number of white specks on the seafloor as well. These are a type of single-celled organism called foraminifera. Some of them are quite sort of globular in shape, rounded, and scientists can use them for a variety of different kinds of analyses, not only understanding the community that's living down here, but also the hard parts. Those white tests record the chemistry of the environment that they were formed in. So they provide a great record of that chemical history.
Cool.
Let's see here. 133 UTC.
Yeah,
So we
shark
have
tooth
about
just below the
40,
lasers.
a little under 40 minutes. Well, that's without setup off bottom. So, but I'll give you like a 15-minute notice. Is that okay?
Oh, it's a shark tooth. No need to zoom unless you want an image. Cool.
Looks like we're getting close to needing to scoop a sample.
All right, well, let's get it here, and we'll, if you want to settle, you can take the shark tooth as well as some nodules. Oh,
Go for now.
We can go somewhere else.
Set it up on course I. So that's probably your last move, pilot.
Yeah, they have to keep up with Sirius as well. So the ship and the Sirius and the ROV work in tandem. Okay.
If you can find an area without nodules, you're doing very well down here. The best thing is to put the push core where there's a very small nodule, obviously, and hopefully push stuff out of the way.
Looks good.
Sounds good. Let's give it a whirl. So do we have time for coring and scooping? Okay. Yeah. Yeah, this same process as yesterday. If we can insert two and then recover them one at a time, that'd be great. Hey, video. Yes, we're doing two for one special. Yay, good job. Nice job, Caitlin.
Hey, Copilot, I'm losing D2 in your Sirius cam. Thank you. Yep, will do.
We can.
Great. Yep.
That is good. And then Levi, when you grab the second one, you don't have to let go of it. Just insert it. If things are good, we can keep going.
That's full white on both.
They'll be counted as two samples, Nav. That's good right there.
Nice.
Yep. Nice.
G?
Yeah, G. And it was this one.
3.
Yep. Yep. Good. Oh, that's nice. Very nice, pilot.
Good. That's great.
Perfect.
You're going to get your nudge.
Give it a little squish in there.
All right.
Those are good cores.
Great.
Nice.
So how much time do we have left, Nav?
We have 27 minutes, 15 of which will be used to set up.
Okay, so we have about 10 minutes.
Yep.
Did you want to keep looking for biology, or shall we just take a few scoops while we're here?
I think scoops while we're here would be great.
All right, you see those two? Okay.
A wee little hop.
Yep, no problem. Well, your mission, if you could please try and fill those geoboxes with nodules, please. The more irregular and gnarly, the better, but don't be too fussy. Anything you can grab.
Gnarly.
Pedal to the metal.
Yeah, the biggest you got.
Ooh, that's a pretty big one.
I think Levi has a great radio voice.
Oh yeah?
Yeah.
Can you sing?
Levi?
Yeah. Can you croon?
Does he sing? I don't know if he sings.
Wasn't there a cowboy who used to sing when he rode his horse?
Oh, what do we
yeah,
got there?
little holothurian. Yeah, that's right.
Bye-bye, Holothurian.
Bye-bye.
Yeah, throw it in with a nodule, sure. Yep. Yeah. See what it's like.
Are you going to try the port box? Oh, right on. Okay.
Yep.
It's got the little door on it. Right on. Okay.
Other one. What's that?
Another holo?
Yeah, it's another holothurian. We'll just take it.
Oh,
what's on the right? On the right's a star.
Like a sea star.
Sea star on the right. Yeah.
Yeah.
Do you have a preference?
We don't have a sea star.
A sea star would be good.
Yeah, if you can. Those nodules in the sea star are the ones. They're much nicer.
We need to get bigger rocks too.
No, they're good nodules. Thank you.
It looked like it might have been an anemone too. I don't know. Geo.
Yeah.
Geo.
Yeah.
Geo with a star.
Geo with a yeah.
Might just get an accidental hitchhiker.
Yeah.
Nav? Yeah, say again. You said the vessel
Yeah.
seems to be doing.
Oh, isopod, decapod.
What? Oh, is there
Actually,
something
is there any way we
Cross.
can hold? We're just taking a sample right now.
Yeah, yeah.
You
Thank
can see
you.
the shadow.
Yeah, no, it went past.
You can see the shadow in that camera.
Oh, we get to try out the port side box with the door. I don't think we've collected in this yet.
Yeah, we collected that holothurian.
In the
Oh, no.
rock box?
Oh, no, not in the rock
Yeah,
box.
yeah. I think this is a first time for this mission. It's custom made. Nav, last time we checked in, we covered about 220 meters or so of ground.
Let
Can
me
you
check.
give us a new calculation? Thanks.
We have gone 226 meters.
Okay. Perfect. Thanks, Nav.
Made an extra 6.
Woohoo! I'll take it. Wow.
Nice, Levi.
Skills. Mad skills. A little extra mud's not going to hurt the push core sample, by the way. So.
And are we counting this as one sample even if we take multiple scoops?
Yeah.
Yeah.
Multiple scoops, aye. Not anymore.
If you've got time, you just keep scooping till we've got to stop.
Was this in Port Ford?
Yeah.
I don't think you can get too many rounds. Yeah. You can always drop that extra weight. Oh, then we lost 2 of them. 3 of them.
Oh.
You're doing great, sweetie.
I'm just going to put samples that
Yeah, just fill
are
it
in
up.
both boxes at this point.
You should offer a reward, like a rubber duck per nodule or something like that.
There you go. So they're mostly going in port forward.
Mostly.
Is that right? Yeah, very good. There you go. Port aft. Okay.
Aft, I think.
Hopefully you'll get some
So
fragments
we're
of the sea star.
technically out of
Yeah.
time. We have about 15 minutes till we need to be off bottom.
One more scoop, drive forward, and then get some water.
Might even be easier to hop.
Oh yeah, drive
You
forward.
have a nodule in mind?
Cool.
Ooh, that's a big one.
You can go for that. Yeah.
Yay.
Yeah, are we taking Niskins on the way out?
Yes, sir.
What's the interval?
Good question. Let me get back to you on that now. Thanks.
Thanks.
So we're going to stow the door on the port side rock box, seal up those samples so they're safe for the return. And then we're going to collect a Niskin sample after we do a little car wash and then start ascending. So
Huh? Come again? I've got a call Bev. Sorry, hang on.
nice. So Rima, can you do me a favor and click on the, yep, and then dive 11. And then review. We're going to take a look at replay view. I'm going to take a look at where they collected the water from before. So go down to 11, 3, 3, 2, 1. Okay, thank you.
Thanks, pilots.
Thanks, front row. That was awesome.
Yeah, great.
Nice suite of collections. Go to C-Log for vehicles. And now you're back in business.
Athena.
Go for now.
It'll be sample 10W.
Yep, you should be good now.
Pulled it. Called into existence.
Can we try and go somewhere a little bit more to clean? I can see the Niskins in view right now. Just trying to get a little cleaner. Close to bottom. But you can let me know if it's going to be too tricky. It's looking better. I'm ready.
Ready?
Wait a minute. You're good. Got it. It's just the word fire, fire, fire made me crack up. It's like, wait a minute, who's listening? There's not really a fire on board. We're just tripping the Niskin, everyone on home. That was good. Thank you. Release the Niskin. All right, Nav, we have some depths for you if you didn't hear Chloe already say them.
Yep. I got him.
You got them already? Cool.
5345. Yes?
Yes, depths for the upcoming Niskins.
Oh, no, I don't have those.
Okay. The remaining ones will be 4,000, 3,000, 2,000, and 1,000.
Oh, sounds good.
Okay. Nice. Good dive, everybody. That was very, very productive. And I think if all the Niskins collect properly, we will beat our sampling record.
What was the previous record?
13.
Oh, nice.
Yeah. Thanks again, front row. Chloe
Nav.
and Rima are going to take over water sampling, and
Steady
I'm going to
up
go set up in the lab. Thanks, everyone, for a great dive, and thanks, Shoreside, for tuning in. Have a great rest of your day.
Bridge, nav. ROV is off bottom. Yep. Nav. Copy. We're going to be doing every 1,000, so not the 5,000. We're going to do it 4, 3, 2, and 1.
All right, 4. Thanks. I was not on extra V that whole time. Oops. Sorry, video. All right. What are we talking about? I was thinking kill flax. We're pretty stable. Yeah. No, that's okay. I'll turn it back off if no one else.
Nav? Yeah, so we're going to be doing them at 4,000, 3,000, 2,000, and 1,000. Yeah, no worries. $18.32 for a cent. We're yeah, it's pretty good though. Your scoops. Yeah, you'll probably make up 2 or 3 minutes on a cent. I mean, I'm not. 2 minutes doesn't matter. It matters to me. Man, is it kind of toasty in here? I think this hoodie has to go. Why did the boat suddenly get hot? What am I missing here? Yeah, I believe it. Yeah. So it's 10 minutes. So it's I timed it. It was like 11. Yeah, so that's why every time we move, like the whole thing takes about 20 to 30 minutes. Yeah. I mean, I kind of did that today, so it seemed to work. Feels much better without the hoodie. Well, I didn't know it was 80. My room was freezing. Yeah, that's true.
Sarah, no way. Sarah, you look so young. No way. I need to see if I can't remember if I've seen you in the sunlight now that I think about it. Oh, wait, I have. 4,000 meters. Copy that. Sample 11W.
Life slaps us. Yeah. Oh, 16 knots. Not bad. Okay. 18. 19. Okay.
Check, check. Am I on the live stream, Caitlin? Oh, hello. Yeah, to everybody watching, we are ascending. We're at 3,100 meters. We're coming up. We had an extended dive today at 5,300 meters today. We collected our scientific samples, and we're on the way up. And we're doing more science samples. We're doing a Niskin sample. We're collecting water at different depths. We have a bottle that closes, that collects water at this particular depth for scientific use. Looks like in 3 minutes, in 90 meters, we go 30 meters a minute. So in just 3 minutes here, co-pilot Sarah will maybe not fire the Niskin, but we'll close it. We'll use a better...
Release the Niskin.
Yeah. We'll release the Niskin model so as not to...
Don't want to scare anybody.
Scare anybody with the word
Get
fire.
The wrong idea.
Yeah.
Especially on a ship, huh? Yeah.
Yes. Your valve packs are on, but not enabled. I think you're aware.
Correct.
All right.
Yep.
Okay. I'll let you take the con.
All right.
Cameras are on, so you can... Ooh, I'll turn the lights on for you, video. Back there. I'm going to give you some polls. It's going to get brighter. Bam. Oh, excuse me. Bam.
Oh, we can switch
Right,
again.
monitor
Oh,
ROV aft. There you go. It's in color mode now because there's enough light for the camera. There we go. So we're looking at we got, what, 5 bottles back there. 3 of them are still in the open position. And we will close number 3 at just the right depth.
3 at 3,000.
And they're designed so the water passes vertically through them like an open tube. So we collect the water that actually is at 3,000, not stale water stuck in there. My fiber numbers up. Side focal. Which sound say the worst fiber was? 14 point?
13.4 something?
Okay, I'll look. I see that. 13.4.
Ish.
That's what I saw too. 13.3.
1 minute till Niskin.
T-minus 1.
Awesome.
Video is at the ready. Nav, you ready for your waypoint target drop? We also need to record the coordinates of this
Watch lead, are you ready?
to add with the metadata for the
Watch lead
sample.
is ready.
Awesome.
They'll record latitude, longitude, and depth. I believe they will run it through like an eDNA filter system, but it's out of my wheelhouse.
This will be the most samples
Alright,
we've collected
10 meters out.
in a dive.
9, 8,
Oh, you can do the countdown. You want to do it? Go. Do it.
6, 5, 4, 3, 2, 1. Mark.
When did it want to go?
Did it close?
I saw it. Just do it again. Do it again.
Okay.
Reset fire. Try again.
Resetting and fire.
Okay. All right.
Okay. So we got number 4.
I think that was
Niskin
teetering.
number 4. Number 3 was failed.
3 was
Niskin
a failed
4
project.
is 3,000
Compromised,
meters.
yeah. I saw it like
I
hesitate.
know it like jumped, but
It
then
jumped,
it didn't go.
but it looks like it's like in a
Kind of
interesting.
weird, right? So how would it get?
I don't know how it gets stuck like that.
Huh.
And that was Niskin 4 at 2,995 meters.
Correct. Yep. Niskin 4.
Always
2,995
keeping it interesting out here.
meters. Okay. So we need to look at Niskin number 3 was failed.
That's a good question, Nav. Give us a minute and we'll get back to you.
Ah, did it close now?
Yeah.
Sure did. Okay.
Mark it.
We will have And Niskin also at 2961.
Let me put an asterisk by it because I don't know if the bottom closed.
Okay.
You know what I mean?
Yeah. So that will be Niskin number 3.
Sorry, watch lead to pilot.
Yes.
Did you just say we had a Niskin at 2961?
Yeah. Talk to Copilot. She's got the depths written down.
Yeah, so we had number 3 failed, went to number 4, we got that 2995, and then 3 ended up releasing after that. So we have a Niskin number 3 at 2961 is the depth.
Copy that. Over.
What if the bungee is too cold and we need to have a more tighter... We'll check that bungee.
Yeah.
I wonder if we get like a scale or like a luggage scale to like check the bungee tension force. I don't know. Spitballing here. I don't know.
We'll figure something out.
Well, we got one left. What's the... If they only get one bottle left, where do they want it?
Ah, perfect.
Copy. In work.
Good question. Well, we... Yeah. And we can let the watch lead decide. Nope.
Watch lead to nav. Hey, let's go with 1,000 meters for the last Niskin bottle. And also from our end, we are logging Niskin 3 as a sample. Correct. Thanks, team. Correct. Go for video. The bottle fire?
That bottle closed. So just to let y'all know. And it's also just labeled as 12W. So will the next one be 14W?
Correct. It'll be 14W.
So
And
there is no video link for 13W.
okay, that's fine. We'll make a comment in our log too for that. Thanks, video.
It's clearly dinner time. It's got to be dinner time if we're talking about food. Oh my goodness. My stomach is grumbling. Oh, that's not going to work for me. Guess we all get to stay with Caitlin. In your house. Party. Party at Caitlin's house. Oh, fun. Nice. Oh no. Oh, going to Jacob's house next. Nice. Burritos, wow. That sounds like a good sacrifice. Whoa. That's nice. Kobe beef. Nice. Okay, why not? Yeah. Yeah, right. Live it up. It's all on me. I need friends like that. Yeah. But it's fun. Splurge while you can. Live it up. Nice. Wow. Mhm. Wow. Yeah. Yum. Mm. Fatty tuna. It melts in your mouth. Oh, that tastes good. Same thing. Mm. Yeah. Try it. Try it once at least. There you go. Right. No, that's for sure. Where are the sharks? No. Have we seen them on any of the dives on the northern circuit since we've been up here? Or wait, yesterday was our. Never mind. No, we haven't. Oh, okay. They're around. Maybe today. Ooh, that would be cool. Yeah. I'm really digging that tail that's in the video. Some sort of thing flying in the water above the. That's neat. Sort of. Mm. There you go. They have whale sharks there too. You can go see.
Copy that. Thanks, Nav. Oh, yeah. Back row is ready for sample 14W. When you're ready, video. No worries. getting close video. Correct. 914 meters per sample 14W. Copy that. Watch lead to pilot. This is back row signing off for the dive. All the best with the last 1,000 meters.